Ladies and gentlemen, welcome back to the Cardiovascular Sciences Cutting Edge Technology Symposium. We'll now begin session 3, and this session is about cardiac disease, and we are honored to have Professor Zhou Bing, co-chair of this symposium, professor at the Chinese Academy of Sciences, and Dr. Alan Poon, assistant professor at the School of Biomedical Sciences, the Chinese University of Hong Kong, as the moderators for this session. Please join me in giving a warm welcome to both of them. A round of applause, and the stage to you.
Introducing the next speaker, Professor Yibing Wang, who is a professor of medicine at the Duke NUS University. So without further ado, I would introduce him and let him talk about post-transcriptional regulation in cardiac development and diseases, from RNA splicing to translation. Thank you.
Thank you so much. Good afternoon, everyone. It's my great pleasure and honor to be part of this incredible symposium. And today, it's actually kind of hard for me to follow Thomas' talk. And I was actually beginning to think about he's going to talk about the RNA binding proteins, and then I can follow with some of the kind of a branch of our interest for a long time is the post-transcriptional regulation in heart. Again, we know that gene expression actually is a full spectrum from genes to eventual products. Even just if you look at the mRNA being synthesized, and it has to go through multiple layers of post-transcriptional modification. And vast majority of our literature is still focusing on the first step. Here is the epigenetics or transcriptome regulation in terms of gene up and down regulation. Yet much, I would say, the activities actually happen here after the mRNA being processed has been, I would say, poorly studied, and their implication in the disease or in the development still need to be explored. And so just from a very simplistic view of the RNA processing, we know that the mature RNA, once it's been synthesized, and it will be processed and through several important steps, including within the nuclei. First is the splicing and some of the modification. And once it's been transported outside of the nuclei, they will actually go through additional modifications in the translation and degradation and so forth. So we're actually interested in many of those steps, and I'll share with you today is mostly in the RNA splicing, but hopefully also touch upon some of the translation studies. And so this, first of all, this work actually was initiated at the beginning by a graduate student at that time, and now she's an assistant professor at the University of Cincinnati. We really try to understand what is the landscape of RNA post-transcriptional regulation, especially RNA splicing, and then how it's being regulated and how it's important for the disease mechanism or the development. And so easier work at that time, this is more than a decade ago, and I still remember those days how difficult to just generate the RNA-seq data. But nevertheless, we immediately realized that the RNA splicing events actually is very prevalent, and unfortunately I skipped this too early, but and from the hundreds of RNA splicing events, we actually identify some shared potential upstream regulators. And particular one is this RBFOX1, because when we see in the disease state, this is one of the few RNA splicing factors actually are significantly modulated or depressed in the disease state. This is a mouse model. This is actually human. And then when we knocked out, we also see, based on this RASO-seq technology, to identify just the RNA splicing events. You can see that in the diseased heart, many of those splicing events actually can be exaggerated by the overexpression, the knockout of the RBFOX1, but then can be also reversed by the reestablished expression of the RBFOX1. So we know that RBFOX1, at that time we estimated, constituted almost 40% of the RNA splicing events that we can detect it in the mouse heart. And functionally, so because this is already published, so I'll go very quickly, that if you knocked out to simulate disease state, you can see the function is deteriorating in response to a pressure overloaded stress. And this is more than just function, but also pathology and the markers as well. But then if you want to understand what is the downstream targets, and we actually did extensive sort of RNA splicing mapping, and interesting enough, we found that one of the downstream pathway is this MEF2 transcription factor. And within the transcription factor family of the MEF2, they all share this alpha-1, alpha-2 microexon switch. And this is a very tiny exon. It's only 15 amino acids, so it's about 45 nucleic acid exon. And it's mutually exclusive, so either alpha-1, alpha-2 will be incorporated into the final product. And we see the change of splicing variant or isoform switch during the disease. And we also see that by virtue of expression the RBFOX1, you can actually modify them. And we have reported assays to show that those are direct targets of RBFOX1. And then functionally, we also demonstrate that these two isoforms actually exert very different outcome in terms of the cardiac physiology, hypertrophy, and so forth. So this is one of the first indication that the RNA splicing events actually contribute to the pathogenesis of a heart failure in the native sort of stress response scheme. And the target, at least in part, is this critical transcription factor MEF2. And follow this study, we also look at the similar sort of RBFOX1 expression in another model of heart failure, that is the post-MI heart. And we can see the loss of RBFOX1 is also consistent in this model. This is in vivo, and this is actually in vitro by oxidative stress in the cardiomyocytes. And we can also demonstrate that by virtue of re-expressing the RBFOX1 using the AAV9 factor, we can reduce the cardiac dysfunction, and as we demonstrate here by the ejection fractioning, but also reduce the remodeling and the cell death. And so here is actually the pathology as well as the marker genes. So we want to know what is the underlying mechanism, and so in the isolated cardiomyocytes, we can reproduce the similar sort of cardiac protective effects, understanding that it's probably involving cell death, at least in part, contribute to its protective effects in the post-MI heart. But what is, again, the downstream target of RBFOX1, whether it's still the MEF2 or others? So we did extensive RNA-seq studies from this in this setting of hypoxia and post-MI heart with or without RBFOX1 expression. And we identified a few that's very interesting target that we didn't see that from the pressure overloaded heart. That is these sort of some of the calcium and potassium channel proteins, but also a very interesting molecule that's the muscle blind LIKE-1. This is well-known regulator, another RNA binding protein itself, but involving the cardiac remodeling, mostly in the fibroblasts that's been reported, but not in the cardiomyocytes. Not only that, when we look at what's the specific exon that implicated in this process, that's quite interesting. This is actually the exon 7, and turns out the exon 7 is also a microexon. It's very tiny in size, and so it's in the sort of a flexible domain of the protein, so we don't know what is the substrate or the conformational impact. But we use the AlphaFold, we can simulate that this insertion actually can have a profound impact on the global sort of structure of this protein. And we can demonstrate that by virtue of overexpression or downregulation. Here is the sRNA. This is actually the overexpression. We see direct impact of the RBFOX1 or its exon 7 inclusion exclusion events. And then using this antisense oligo, you can actually target the exclusion events of the exon 7, and so independent from the upstream activator RBFOX1. And we can see that just by virtue of changing the splicing events of this exon 7, you can actually alter or provide either exaggerate the cell death or protect the cell death in the cardiomyocytes. And in vivo, we actually see similar outcome based on the echo post-MI heart as well as the remodeling, that the antisense oligo that can prevent sort of the inclusion of this exon 7 turned out to be protective as well. So suggesting that this muscle-blind microexon 7 really play a very important role that we have not appreciated before about the cardiac protection. And so here is actually our hypothesis, and now we're testing this indeed in the more long-term as well as additional sort of a preclinical model, hoping that this potentially can be utilized as a therapeutic modality, because again, using the antisense oligo is actually quite effective in delivering to the heart and as well as changing this particular splicing events. So we talk about the RB-FOX1. You can see that by downstream effects of the specific splicing can have a pretty strong impact on the pathogenesis of the heart failure, whether it's a pressure overload, induced the cardiomyocyte hypertrophy and the heart failure, or post-MI heart. And what I want to also show you is the impact on the translation side. So this is actually the same concept that we actually observed among many other RNA splicing events, is that the disease, the heart change actually reflects or mirror what we see during the cardiac maturation. So this is done actually by JJ and Josh Lee from our lab, and identified that in the neonatal heart to adult heart maturation process, in addition to the metabolic change, in addition to the cell cycle regulation, we always see this very strong signature in RNA processing and particularly splicing. And turns out RB-FOX1 is also highly induced during the maturation process, correlated with its exit of the cell cycle as well as functional maturation. So we showing that in both cultured neonatal myocytes as well as human iPS cell-derived cells, if you express the RB-FOX1, you can actually promote the cell growth in terms of size, and also promote the gene expression that associated with maturated myocytes. You can increase the contractility, and you can actually even improving the sort of calcium cycling effects. And so here is actually the control. Here's the RB-FOX1 expressed cells, and eventually the electrophysiological maturation. So it seems like a very potent regulator for cardiomyocyte maturation. And this feature actually repeated many times that we saw the RNA splicing turned out to be a signature in the disease state, at least is a signature of a so-called fetal-like reprogramming. Okay, so we talk about the RNA splicing, and what I want to also share with you is a recent story that unpublished about the translation aspect. And so this was done actually in a kind of accidental way in terms of its discovery, because we at that time working with Professor Jack Lucis at UCLA, using this sort of assistant genetic approach to screen for hundreds of strains of mice, actually looking for the phenotypic outcome in response to pathological stress. And in this case, it's the chronic iso treatment, beta-adrenergic overdrive, and looking for all features of heart failure and fibrosis, and you name it. You know, we're still actually going after many of the findings today. But one of the features that we saw was the gene that associated with the growth of the adrenal gland. And so this is actually the one hit that we saw a quite significant impact on the adrenal size. And also within this genetic locus, we saw the gene that within this loci actually expressed very highly in adrenal gland. And so this is the EPRS protein. Right, so you can see the expression. This is in the adult heart as well as we see similar features in adult human tissues. So what is this protein? And also the reason we are interested, of course, we know that adrenal gland function is very important in the crosstalk with the heart to maintain, sympathetic tone and overall stress response. And so that gets us to very curious about the role of this EPRS protein in this crosstalk between the adrenal gland and the cardiac, especially in the pathophysiology, especially baseline physiology as well as in response to stress. And so what is the EPRS? And this turned out to be another enzyme. It's the tRNA synthesis that actually charge the tRNA for either proline or glutamate. So it's one of the essential protein synthesis enzyme. However, in the literature, we actually knew that this particular protein is more than just the enzyme. It's also an RNA binding protein that has very interesting features of protein translation control. And however, in the interferon response pathway, the EPRS is actually a very potent translation suppressor. This is part of the core units of the gate complex, is so-called GAIT. So this is a protein that can uncouple the transcription from translation to dampen the interferon gene regulation. And so we don't know what is the EPRS function here in the adrenal gland, whether it's actually very important for sort of the protein translation in general, or it actually has a particular regulatory mechanism. And so, as I said, EPRS is highly expressed in the adrenal gland. This is, again, the mouse tissue, and it's correlated very well with the catecholamine synthesis enzyme, obviously. This is tyrosine hydroxylase and PNMT and so forth. And you can see that in the disease state, this is a post-MI, and we actually see in the hypertensive disease models, and the EPRS in the adrenal gland is actually overexpressed and correlate to catecholamine surge as well. So when we perform the adrenal gland specific knockout of the EPRS protein, and we see first is the adrenal gland, the medulla structure. This is where the catecholamine is synthesized, actually markedly reduced in size. And also the catecholamine production is actually reduced as well. And then what we also see is the enzyme that is responsible for catecholamine synthesis. Here's the pathway from tyrosine all the way to epinephrine. You can see that almost many of those key enzymes actually are reduced at the protein level. Yet, if you look at their mRNA level, they're okay, right? So this is the first indication that we know it's probably important for protein translation. Of course, you will ask, since this is a EPRS, is a part of the protein synthesis, you probably inhibit the entire protein synthesis machinery, so no surprise that you inhibit those enzymes. So to our surprise, what we saw is that even if you knock down, this is actually the knockdown of the in the PC12 cells, and we also did this in the whole tissue. You can see that protein synthesis, either the contents or the protein synthesis rate based on the pure myosin labeling, there's no impact. So the protein synthesis was actually maintained even without the EPRS expression. And yet, the specific enzyme continued to actually being blocked. You can see those targeted protein. And if you overexpress EPRS, you also do not see a significant impact on the protein synthesis rate overall, but yet you actually promote the specific protein synthesis of this target genes. So we at that time realized that maybe this is a enzyme independent activity for protein regulation. So indeed, what we have done is we found that the EPRS actually as a function as a RNA binding protein. It interacts with the 3' UTR of the target gene. This is just by the enrichment over the threshold. And you can use a reporter construct to use only the 3' UTR to look with the luciferase, and you can see that it's response to EPRS overexpression, but not in the reporter the control reporter construct. And this induction is actually independent from the enzymatic activity. You can mutate. This is a human mutation associated with the tRNA synthesis activity. So it's an enzymatic independent activity. You can see based on the luciferase induction. So all those line of evidence suggests that this is actually not just the enzyme for tRNA synthesis, but actually an RNA binding protein for protein translation. And so here, the additional evidence I thought is really nailed the sort of evidence is actually in vivo. We can use the trap model Professor Bill Poole actually shared with us, that we can target the ribosome in the adrenal gland and then pull out the ribosome and looking at those mRNA, targeted mRNA on the ribosome loading. And you can see that we can enrich those ribosome with the transgene, and then specifically looking at the mRNAs associated with the ribosome. And without EPRS, the ribosome loading of those targeted mRNA is actually significantly impaired. And so since adrenal gland is very important for cardiac homeostasis in terms of function, and we look at the functional outcome, and lo and behold, as we expected, if you have lower level of catecholamine, the baseline contractility was decreased in same as the heart rate. Yet this phenotype is a physiological in the sense that you can just infuse with dopamine and completely rescue this phenotype in terms of contractility and heart rate. And this is not heart failure. If it's heart failure, actually you would not observe this because they are usually a adrenergic sort of a resistance in terms of their signaling. And however, when we do the post-MI in those animals, what we observe is actually the knockout animal has a much better preserved function in the long run, and suggesting that the overdrive that often associated with the post-MI disease state turned out to be actually quite detrimental, and it requires the EPRS mediated catecholamine synthesis in the adrenal gland. And just highlight the importance of adrenal and heart crosstalk through this post-transcriptional mechanism. And we have additional data to support, the molecular mechanism why it's involving the ribosome loading. Okay, so this again shows our sort of efforts to highlight the importance of mRNA post-transcriptional processing, whether it's a splicing or whether it's regulating its ribosome loading in translational control. And as we know that this is only very few examples of the very complex sort of a mechanism, and we believe that the post-transcriptional regulation is really an area that probably will yield many of surprises as well as interesting insights. So with that, I already highlight the contribution, and this is ongoing collaboration with Dr. Chen Gao and our colleagues from the lab, and I'll want to leave this for the audience because we're hiring, and for the senior investigators as well. Please, if you can transfer this information to your lab members, really appreciate it. Thank you.
So thank you so much for a very interesting talk. Any questions? Actually, I have the first question. So relating to the maturation work, I'm interested. So you've shown that RBFOX1 is important for cardiac maturation. So how does this protein respond to protocols that promote maturation, like metabolic selection or electrical pacing?
Yes, so in every model that we examine their data, we always see the RBFOX1 is expressed parallel to the maturation process. We actually had an independent project that we identified the upstream regulator for this particular gene expression as one of the upstream enhancers, and we actually even made a reporter mouse model and showing it's even in vivo it shows correlation with postnatal maturation process. Yes.
Thank you so much.
Yes.
Actually, two questions, really very nice. I mean, first, also related to maturation functions of of VII. So, I mean, you showed very nicely, I mean, that MEF2 switching, but other direct targets, I mean, we also have, you probably referred to that, some RNA binding proteins that are necessary to generate the more adult, mature isoforms of certain genes. But in relation to your gene, is there also a lot of target genes?
Yes.
More the mature form in addition to MEF, and is that the combination that does
That's
it?
right. That's right. Definitely, we actually highlight only the MEF2, but there are hundreds of downstream events altered by the RBFOX1 expression, and we actually did so-called the RIP-seq, try to identify the all downstream targets. There are many actually. Yeah, a lot of them are metabolic, and a lot of them are also sarcomere.
Yeah, might also be an interesting means, along the same line that I proposed. But second question, I mean, this loading of your targets in the last part, I mean, you showed binds to the 3' UTR, and then you have enhanced loading to the ribosome. Any idea how that worked?
Yeah.
How it facilitates recruitment?
In the interest of time, I skipped the kind of a molecular studies, and so we actually did the very similar approach as you have done with IP mass spec, and we identified one of the binding partner is the elongation factor, and it is a classic factor required for ribosome loading efficiency regulation. And so it's kind of interesting that this protein somehow become a partner of this sort of translation initiation complex machinery. And it's exactly opposite from its role in the gate complex, which is actually blocking the translation. And this one is promoting it, but it's in a cell type specific manner. Yeah.
So, any more questions? So thank you, Professor Wang. Thank you so much for the talk. So thank you very much. So now I'd like to invite our next speaker, Professor Randolph Wong. So he's the Professor and Chief of the Division of Cardiothoracic Surgery in the University of Hong Kong. And he's going to talk to us about innovation in aortic diseases, diagnosis, and treatment. So thank you.
Right. Well, thank you, Ellen. Would like to thank Kathy and Bean again for having me here. It's my second time in the Cardiovascular Symposium, and give me great pleasure to understand that how much a surgeon doesn't know basic science, and yet we are still practicing surgery every day. And so I'm going to show my ignorance in the coming 20 minutes. So my disclosures here. History about aortic surgery. Actually, it starts from around 1959. The first operation, aortic surgery, is actually done by American surgeon Denton Cooley when he passed by Hong Kong, go to Beijing, and then he operated on an aortic patient. And then ever since then, 20 years thereafter, it has been the era of the British colony, and then a lot of surgeons in Hong Kong were trained in UK or Australia and come back. But yet, the early report published by Dr. John Leung that our aortic surgery having a mortality of 25%, okay? And especially aortic arch surgery, which replacing the aortic arch, the mortality is 100%. So imagine. So basically it's like going to one-way ticket, having a one-way ticket. The reason is when we replace an aorta being like a highway train that, bringing oxygenated blood from the heart to the rest of the body, it can't be stopped. And if you need to replace the arch, you need to stop the circulation to the brain, and you can imagine what will happen. Now, but the subsequent 20 years, the year that I joined CHK and after graduate from the med school, that we did make some impact on this overall outcome. So, in fact, we introduced something called hypothermic circulatory arrest. We bring down the body metabolic rate. Of course, we didn't look at the metabolomic at the time, but by believing that actually we reduce the temperature, reduce the myocardial enzymatic activities, and then we can protect the heart, protect the brain, protect the rest of the body organs. And thereafter, we also introduce hybrid intervention, endovascular intervention that do not require cardiopulmonary bypass. And we are able to improve our outcome. And more recently, we have the instrument of the hybrid theater complex aortic arch surgeries, introduce it, and now we go to minimal invasive aortic surgery as well. So actually, it's all about the quality assurance. It's very important that we, as a surgeon, we don't know much about the basic science that you just mentioned. I will try to look into each of them carefully about the T-cell, everything. But we do need to look at our outcome, about the patient survival, patient complication. So from year 2006, we collect all the cardiac surgical data outcome, and there's over 7,000 patient data now available that we do have a clinical team as well as the research team sitting over there to support me today. And now aorta, I think we got covered that. It's a high-speed train that bring oxygenated blood to the rest of the body vital organs. When there is a problem of the aorta, that could either be aortic aneurysm or aortic dissection. Now, just now I have a talk with Professor Zhao Hui. In fact, interestingly, when the Nature Communication published their GWAS data, actually descending and ascending aorta were coded by different gene, okay? And it's because they have a different embryological origin, because I think the ascending aortic roots are from the ectoderm, the cardiac crest cell, a neural crest cell, and the descending are from mesoderm. So that's why, interestingly, Stanford in year 1970, they divide Stanford type A, Stanford type B aortic dissection, because there's a naturally occurring cleavage between the joining of the mesoderm and the ectoderm right here. That's why I don't know whether Stanford, they understand there's such a thing, and then they give a Stanford type A, type B. But having said that, aortic dissection, silent killer, a lot of patients have aortic aneurysm that they don't know. But once they develop aortic dissection, the mortality is very high. So these are the two main complications, including dissection or rupture. And with the presentation, 40% of the patient die at the scene, and 20% more die before surgery. And as you know, even the most updated data, the operative mortality is still hovering around 20%, because a lot of patients already have malperfusion, or a lot of patients already have ischemic insult. That's why I think it's very relevant, a lot of the talk that we try to reduce the ischemic insult to the body. So, and more importantly, a lot of the patients after the operation, they would still need to have, even if they survive this horrible thing, they still have the remaining aorta to worry about. Some of them might need a second stage operation. But having said that, we can see here the overall aortic activity increased from 20 years ago, around 13 cases, to last year, 130 cases. And the mortality, we are able to reduce it from 20-something% to now around 5% mortality for type aortic dissection. And that is a lot of work that's been done clinically in terms of clinical research about organ protection, about brain protection, about the setup of the cardiopulmonary bypass. We also look into not only the immediate survival, we also look at the 5-year survival, and found out there are other factors that would govern the more midterm to long-term survival. And this is actually last year data showing our elective aortic mortality is 0%. And with that, we are able to reach to the HA Outstanding Team Award and have multiple press conferences. Thanks to the hybrid operating theater, as I said, ultimately, if we can do all the repair without cardiopulmonary bypass, maybe that would reduce the systemic inflammatory response, reduce the cytokine storm. That's why there are better way is to using endovascular approach. That's why in 2012, we established this hybrid operating theater, and hopefully using this endovascular treatment, we can exclude the aortic aneurysm or treat the aortic dissection. And that is minimally invasive approach. And because in the past we need to have a hand neck vessel bypasses, that definitely would change the body blood pressures to a lot of this patient. But now we can use a percutaneous approach. So the whole procedure can be done with only 8mm wounds over their groins. And in the recent year, we also introduced these. We are the very first adopter of this device is the aortic arch branch graft. So we don't even need to have the hand neck vessel bypasses. We can use endovascular, and then with these double wire technique, we can introduce this side branch and cover and repair the aortic arch with this total endovascular approach. By doing this, the blood mainstream of the blood is being excluded to the aneurysm, and the aneurysm will regress in size. So a lot of patients can be discharged in like 2 to 3 days without major bleeding. So we are also first in Asia to perform this total arch frozen elephant trunk hybrid approach. And we were first in Asia, and then we have a press conference about the first 25 cases that we have done. Now we have done 70 cases with nearly 0% mortality. And so imagine we were bringing the total aortic arch replacement from 100% mortality, like 20-something years ago, to now nearly 0% mortality with these advances of technology. And I want to skip this. So these are the publications related to the different aortic intervention that we have done. And this is actually red hot. We just presented the multi-center trial that I am the PI to lead the Asia-Pacific region, including, I think, Korea, Thailand, Singapore, Malaysia, and Australia, and into this multi-center trial of the total arch frozen elephant trunk. Just to give you a benchmark, in this study, I'm also part of the European study. The mortality of total arch frozen elephant trunk in the European study is around 5%. In American study is around 7 to 8%. And this is very remarkable because we recruited over 114 patients, and with this kind of total arch frozen elephant trunk devices, and in our cohort, we have a mortality of 1.8%. So that's real life. I mean, these are all genuine, validated data by a third-party clinical trials company to validate all our results. So actually, we are achieving really good clinical outcome with this, with minimum renal dialysis, minimum complication. Now, as I said, these are all the patients that are actually able to go to the operating theater. The key thing is, if you remember the first pie chart, once the patient develop aortic dissection, actually around 40% of them die. So the key is, can we screen patients before disease happen? So in fact, we have that incentive is we use the point-of-care ultrasound system to screen all the hypertensive patients, and we found that over 1,529 patients that we screened, the prevalence of thoracic aortic aneurysm is 7.5%. A majority of them are elderly male hypertensive patients. Now I want you to remember this because I mentioned degenerative change are mostly aneurysm, okay? And they are usually older and male, smoking male. So I'm at risk. And we have that published in Annals of Thoracic Surgery, and male gender age over than 65 are risk factor for degenerative thoracic aortic aneurysm. And we now collaborate with the CAIR and to work with the algorithm that could be automatic measurement by an ultrasound. By reading the ultrasound, there will be automatic acquisition of the keyframe and then measurements. Because the key problem of screening with ultrasound, point-of-care ultrasound, is the expertise, lack of expertise. We don't have enough echo nurse expert enough to screen all our patients. So with this, we have the idea of having AI to help us to give a measurement, and we are able to achieve very high agreement between the AI as well as the cardiologist. So Alex Lee is part of the project, and we are now applying for a patent about this algorithm. And ultimately, what we want is to have a fully automatic robotic arm that we would place at the A&E. And if anyone have chest pain, we just ask this robotic arm to do an automatic keyframe and then measurement. Okay. Another thing, as I mentioned, prediction and prevention. As long as you can prevent and predict patient who is going to have problem, then it's better. Now, one thing that I just mentioned is a lot of patients with thoracic aortic aneurysm, they are older. On the other hand, we are seeing a lot of younger patients who develop aortic dissection, even when the aorta is not dilated. So you can see for aortic dissection, 25% of our patients are actually younger than 50-year-old. whereas for aortic aneurysm, 10% less than 50-year-old. So we do believe there are an important role of genetic predisposition for the aortic aneurysm and dissection, especially in younger patients. So we did a pilot study with the help of our Department of Surgery, bioinformaticians, and these are the pipeline. I try not to pretend I know much about this, but basically the finding is we do find the sporadic, which means when they are younger than 50-year-old, they have similar set of gene mutation with those with family history or known syndromic patient, aortic syndromic patient. But when they are, and these group of patients, they have basically no overlapping gene with those degenerative type of aortic aneurysm. So we are also looking into our own data as well as we collaborate with Hong Kong Genome Institute. Kathy is part of it, together with Professor Ken Song. Now we identify several genes. I think I noted this name appeared many times today because they are the important governor of the smooth muscle cell and the as well as the mitochondrial energy workhouse. So they are very relevant to the aortic disease development. So I think together now we're sending sample to Kathy and we will try to work on downstream and see whether we can have more insight about this. The other thing is with the team here, our team of the AI, so as I mentioned, when the patient come in with chest pain, we want to differentiate whether the patient is having acute coronary syndrome, myocardial infarction, aortic dissection, or pulmonary embolism. So with this, we are awarded with the HMRF project, and we hopefully can have a rapid multimodality screening for patients, including clinical parameters, biomarkers, as well as plain CT4X. So with that, we developed a model. At the moment, we have this model developed that with high DICE index for the measurement of the aortic size at the very site. We are able to make measurement classification as well as auto-segmentation. So this model can automatically give us the 36 annotation, segmentation of the aorta automatically. So now we still rely on the radiologists to give us report, tell us the size, but these program could help automatically generate these report. So ultimately, we also want to predict which patient is going to have problem. Like this is one of the procedure that we have done, total heart frozen left trunk. But a lot of this patient might still have downstream aortic problem. But our program is able to have a longitudinal ability to see whether they will need having any endo leak. Because by looking at the plain scans without contrast is less harmful to the kidney. And with the plain scan, with this AI program, we are able to look at the aortic size automatically and suspect whether there's any endo leak, and then we can intervene at early time to prevent long-term mortality. So these are the collaborators that we have for this project. Last but not the least is robotic magnetic endovascular repair. So although the aortic aneurysm is basically able to use stenting, but to negotiate the wires through complex arch anatomy sometimes is difficult. So we now using a magnetic navigational, using a robotic arm to steer the tip of the catheter and stabilize it. And this is one of the projects that Dean Chiu just mentioned this morning. We are about to do a first in human magnetic steerable wires in human to help us to negotiate through complex arch anatomy. So with these like PS5 joystick, we are able to remotely control the tip of the catheter and direct it to the designated place of body. So, ladies and gentlemen, we believe that we are able to reduce the operative mortality of aortic disease patients. But yet, I think prevention is better than cure, and also we need to have a better understanding about the disease pathology. So I could see a lot of hope here because now a lot of you are spending a lot of time to identify the culprit gene, the mechanism of disease happening. So we believe that with the AI, with robotic, with the advanced understanding of genomic and the cellular level, we were able to make big impact to our patient. So thank you.
Thank you so much, Randolph. Any questions?
Hi, Randolph.
Hi.
Nice work. We know that from embryonic development, the ascending aorta, aortic arch, and descending aortas are derived from different embryonic origins. I think it's from secondary heart field, neurocrest cells, mesoderms. But I'm wondering if in the human tissues, if you have any access to those smooth muscle cells from different segments, and I wonder if there are any gene differentiation, different gene expression of these smooth muscle cells, different segment of smooth muscle cells in the human. You have any data or any access to such,
That's fantastic
tissues?
question. That's fantastic question because we do have access to all these segments. We do have aortic root, we do have aortic arch, descending and abdominal aorta, even carotid artery.
Do you find any?
But we haven't
gene
done the
difference?
downstream, so we are waiting for, we will send tissue to Kathy and then for her to work on this. Yeah. This is at the very infancy because in Hong Kong, I think barely anyone is doing this. A lot of patients now, the thing is, people, even in mainland China and around the world, are doing more and more endovascular. When they're doing more and more endovascular, they don't have tissue. Right? But we are still doing majority of our work with open surgery. But I just knew Kathy like 2 years, so we will collaborate more and then send more tissue to Kathy. Now we have sent around 50 cases. So hopefully by the next culture symposium 2 years later, we will have some data for you.
Thank you.
I actually have a quick question. So as a basic cardiac scientist, I'm impressed with the outcome. So I think in one of the slides, you mentioned biomarkers. So these biomarkers, can they predict or diagnose aortic aneurysm?
Not yet, unfortunately. That's why that's another holy grail that we are looking for, because I think a lot of the protein or gene that you guys just mentioned would be perfect for screening. I mean, if you, I can't name them now, but if they can be make into an aptamer, a rapid test like COVID, so A&E, when they come in, and then we check the marker that been mentioned this morning, if they are relevant, we can see the relative distributions of each. For example, we can test 10 of them, like the DNM3, the CoL, I don't know whether we can make it into a small kit. And then we can see the relative distribution. And troponin, D-dimer. So, of course, pulmonary embolism will have high D-dimer and low troponin. And myocardial infarction, high troponin, low D-dimer. And aortic dissection could have moderate raise in D-dimer, but low troponin, something like that. So, but you combine with several markers, I think if we are able to make this, we will be very happy and rich. Yeah, rich and happy. Right.
Well, I would be glad to know you now and in the future. Okay, so any more questions? Okay, so thank you so much, Randolph.
Our last speaker of this session is Professor Yu Nie from National Center for Cardiovascular Diseases, Peking Union Medical College, VII Hospital. He will give us a talk on uncovering regenerative targets for cardiac repair.
Okay,
Please.
Thank you for introduction by Professor Zhou. I'd like to share our research about uncovering therapeutic targets for heart regeneration. Let's begin with some background. Myocardial infarction, as we know, the leading cause of death worldwide. Every year, over 9 million people die from myocardial infarction. Why it's so lethal? The main reason is the myocardial infarction triggers massive loss of cardiomyocytes. A severe myocardial infarction can lead over 25% of cardiomyocytes to die shortly. Coupled with cardiomyocytes quit the cell cycle and cannot effectively renew. So this damage ultimately drives cardiac remodeling and then heart failure. And recently, evidence increasingly suggests that stimulating endogenous cardiomyocyte proliferation offers a promising strategy to promote heart regeneration and repair cardiac injury. So we believe that heart regeneration is the key to improving myocardial prognosis. Then raise the question that how can we identify therapeutic targets for triggering heart regeneration? First, we need an animal model of heart regeneration to capture its regenerative signals, such as zebrafish, neonatal mice, and sometimes we use adult mice. Because the neonatal mouse heart have the robust capacity to regenerative its cardiomyocytes, such as the heart of one-day neonatal mice can regenerative completely after resecting about 15% of the ventricular myocardium. So we choose the neonatal heart mice apical resection as the model to start the heart regeneration. There are two reasons we chose the animal model. First, it's a mammalian model. Unlike zebrafish, neonatal mice heart can have fully formed four-chamber structure, much closer to human heart. That gives it strong translational value. The second reason is the regenerative response is robust in neonatal heart. Unlike adult heart, the neonatal mice heart can undergo robust regeneration after injury, making it an ideal system to capture regenerative signals. Building on neonatal mouse heart apical resection model, we performed single-cell RNA sequencing of cardiac tissue at 1 day after resection, a time point we believe is essential for initiating cardiomyocyte proliferation and heart regeneration. Based on single-cell RNA sequencing data, we mapped cellular dynamics during heart regeneration. We found that cardiomyocyte re-entry cell cycle and endothelial cell drive angiogenesis and removed a lot of immunocells, recruited a lot of immunocells, and the fibroblasts showed marked changes in their extracellular matrix genes expression profile following apical resection injury. Because that time fibroblasts how involved in heart regeneration is unknown, so we make our attention to fibroblasts. Fibroblasts involve heart regeneration maybe by inducing extracellular matrix remodeling. If cardiomyocytes are the seeds of regeneration, the ECM is the soil that determines whether they can take root and grow. So we propose the hypothesis that cardiac fibroblasts might contribute to heart regeneration via ECM remodeling. To confirm this hypothesis, we extracted cardiac ECM from neonatal hearts 1 day post apical resection injury. We then treated primary mouse cardiomyocytes with this injury-induced ECM and measured the proliferative response. Immunostaining revealed that PA3 positive cardiomyocytes increased significantly after treatment with the remodeled ECM isolated from injured myocardium. P3 is the marker of mitosis, so these results indicate that ECM remodeling is essential for activating cardiomyocyte proliferation. The next question is which ECM components regulate cardiomyocyte proliferation? To answer this question, we isolated cell-free ECM from injured neonatal hearts and analyzed the protein components by LC mass. By integrating these cell-free ECM proteomics data with single-cell RNA sequencing data, we identified 8 fibroblast-derived ECM candidates that were upregulated after injury. To pinpoint which ECM factor drive cardiomyocyte proliferation, we exposed primary mouse cardiomyocytes to each of the 8 ECM recombinant protein individually, then measured the proliferative response of cardiomyocytes. Among all 8 cardiomyocytes, only VII significantly increased both Ki-67 positive and Pa3 positive cardiomyocytes. These results indicate that VII might be the key ECM component that promotes cardiomyocyte proliferation and heart regeneration. Oops. VII is a ECM proteoglycan that acts as a key structure and regulatory molecule in various tissues. So VII needs to bind to cell surface receptor to active downstream pathways. Using Western blot, we examined the activation of its known receptors in cardiomyocytes after exposed to VII, including CD44, integrin beta 1, and EGF receptor. We found that only integrin beta 1 was activated by VII, while no obvious changes in CD44 and EGF receptor were observed. To explore signaling downstream of integrin beta 1 activated by VII, we performed RNA sequencing on VII-treated cardiomyocytes and observed activation of ERK and AKT pathway. And functional validation revealed that if one blocked the ERK pathway by its inhibitor and AKT inhibitor, both of them could block the proliferation activity of cardiomyocytes, confirming that VII drives cardiomyocyte proliferation through both ERK and AKT signaling. The next question is, does VII drive heart regeneration? To test whether fibroblast-derived VII regulates heart regeneration, we generated a fibroblast-specific VII knockout mouse by crossing CO1A2 CRE-ER and VII flux mice. And the CO1A2 CRE-ER mice is from Professor Zhou Bing. Thank you, Professor Zhou Bing. Then we performed apical resection injury in neonatal VII mice. At 7 days post-resection, cardiomyocyte proliferation was blocked when VII was deleted specifically in fibroblasts, as shown by Pa3 staining. These results indicated that fibroblast-derived VII is required for endogenous cardiomyocyte proliferation in neonatal heart during heart regeneration. Next, we measured the regeneration by Masson staining. At 21 days post-resection, Masson staining revealed that VII flux litermator hearts with the expression of VII in fibroblast can the hearts can regenerative completely, no fibrosis, no scar. But in the VII KO hearts, the regenerative capacity was completely lost. These results indicate that the VII is required for neonatal heart regeneration. Then we want to know, can VII as a peptide drug promote cardiac repair in adult myocardial infarction? To explore the potential therapeutic application of VII in ischemic heart diseases, we introduced myocardial infarction injury in adult mice and treated them with single intramyocardial injection of recombination VII after a myocardial infarction. Serial echocardiography showed that VII significantly improve heart function after a myocardial infarction across all doses tested, and injection fraction higher than PBS controls as early as 14 days post myocardial infarction. These results uncovered that VII could improve cardiac function after myocardial infarction. Masson staining showed that VII significantly reduced the infarct size at 28 days post-infarction. These results position VII as a promising therapy to improve outcomes for patients with myocardial infarction. We want to know why the scar size is reduced. So we further reviewed whether VII improved cardiac repair by promoting cardiomyocyte proliferation. We evaluated cardiomyocyte proliferation in injured adult mice hearts, tested with VII at 7-day post myocardial infarction. Stain for multiple cell cycle markers revealed that a signal activation of cardiomyocyte proliferation. To assess the translational potential, we treated human iPS-derived cardiomyocytes with recombinant VII from human. The results show that VII significantly increased proliferation, demonstrating its pro-regenerative function is conserved in human cells. Make a summary. Cardiac fibroblasts contribute to heart regeneration through VII secretion, which in turn stimulates cardiomyocyte proliferation. Second, VII is a novel cardiac regenerative extracellular matrix component. The last, VII promotes cardiac repair in myocardial injured adult mice and enhances proliferation of human iPS-derived cardiomyocytes, positioning it a promising therapeutic candidate for ischemic heart diseases. These results have published in circulation, and the editorial said that how can young extracellular matrix promote cardiac regeneration? VII is a very important extracellular matrix for neonatal heart regeneration and adult mice repair after myocardial infarction injury. Last year, our recent study also revealed that spatial transcriptomics showed endocardial cells also regulate ECM remodeling and contributes critically to cardiac repair. Currently, we are conducting chemical modification and process development studies to enhance the druggability of Verscan. After all this, we are planning large animal studies to evaluate its therapeutic potential in myocardial infarction, and which is an essential step toward clinical translation. Thanks to my collaborators and funds, and thanks to every member of my lab. Welcome everybody to Fuwa Hospital. This is the National Center for Cardiovascular Diseases and the China Cardiology Park. This building is the CK Laboratory Cardiovascular Disease, and my lab is in this building. And this is the vast distinct of Fuwa Hospital. It's a campus with the research building and hospital. This is a cardiovascular farm and medical device industry cluster near to our research campus. So the research could from the lab to bench via in a very short distance. Thank you for invitation from Professor Liu and Professor Zhou. Thank you for your attention. Thank you.
Thank you.
Thank you very much for the nice presentation. I have a question. Versican, to my knowledge, is also induced in adult mice, also in fibroblasts, and it's also expressed by many other cells. So why do you give an added value if you inject it in adult mice? Because they should have it also.
Okay. I got it. Verscan is also in tumor. It could promote tumor cell proliferation, but in heart, it could promote cardiomyocyte proliferation. And Verscan is expression is high level at embryonic heart, but during postnatal heart development, the Verscan expression is reduced very fast.
But it's still among the highest expressed extracellular matrix proteins, in at least on transcription level after myocardial infarction at day 7. So I just wonder, maybe it's on the protein level, but on RNA level, it's certainly very high in adult mice hearts.
I don't know that. Thank you.
Hi. Hello. That was very interesting. Can I ask, in the process of regeneration, the differentiated cardiomyocytes, you say they proliferate in order for repair. Do they actually directly proliferate, or do they do they undergo any change to become a progenitor cell and then go back to differentiate and proliferate? I mean, I am not familiar with the process of regeneration in the heart.
Oh, thank you for your question. I know that Verscan could induce the cardiomyocyte proliferation. It's needed to de-differentiation first step. It's really that.
So if you treat with Verscan, they de-differentiate?
Yeah. We have detected the de-differentiation markers such as alpha SMAR and some other de-differentiation markers. It's really high during the Verscan treatment cardiomyocytes.
And are there any is there present any small population of progenitor cell that's still sitting in the adult heart or the mature heart that gets activated? I mean, what I'm trying to say is the source of the repaired tissue, is it always already from the differentiated cardiomyocytes which are now proliferating, or is there any other by lineage, another population that actually is contributing?
Actually, frankly, we have not not isolated the subpopulation of cardiomyocytes from this, but I found it's the effect of the Verscan promote cardiac repair is significantly. So we but some study have identified the subpopulation to promote endogenous cardiomyocyte proliferation, involve this endogenous cardiomyocyte proliferation and heart regeneration. Some study haven't done it.
And the source of the versicon is the fibroblast, not the cardiomyocyte.
Yeah, just the fibroblast. And we cross the COL1A2 fibroblast specific knockout the versicon. We can find that the heart regeneration capacity is completely lost. Thank you.
Just probably one comment. Yeah, a lot of people looked for cardiac progenitor cells in the adult heart. Yeah, so islet positive cells, but this was all negative. For sure, no progenitor cells left in the adult heart. We can talk later. I just want to ask my question, which is a very provocative one. Yeah, so. I mean, we have some recent evidence that particularly during neonatal heart regeneration, there's also a considerable amount of migration. Yeah, so that neonatal cardiomyocytes can also migrate locally to fill up that defect. So I was just wondering whether you are 100% sure that's only this relatively modest induction of proliferation that contributes to the beneficial effect, or whether by changing the ECM you might also facilitate local migration of cardiomyocytes, so that from intact myocardium, some cardiomyocytes proliferate to the diseased site and then fill up the defect. Did you look at that, whether there is any evidence for enhanced migration instead of proliferation?
It's a very, very good question. We actually do some research about the stiffness of ECM. We found that versicon could make the stiffness as change. So I believe, just like you said, versicon could do something except its signaling role, but also to make the ECM stiffness change.
Just a bit of a challenge here to monitor migration in vivo, so that needs actually some really refined labeling and following migration. It's definitely not trivial, unfortunately. But
okay, thank you.
To see more of that.
Thank you. Thank you.
Yeah, I have one more question.
Okay.
In many injured organs, in addition to the heart, this fibroblast will respond and secrete versicon. Could this versicon promote other organ regeneration, like promote epithelial cell proliferation for tissue repair, similar as in the heart? Can this be brought in other applicable to other organ or tissue regeneration and repair?
Frankly, we have not do some research about other organs, but I think versicon might be effectively to improve other tissues repair, because I think it could, just like Professor Brown said, it could make the ECM stiffness could change. I think it's a very important microenvironment for the cell proliferation and the repair.
Okay.
Thank you.
Okay. Thank you, Niet, Professor Niet, for your wonderful talk.
Thank you, Professor Zhou.
Thank you so much. May I invite our speakers and moderators for this session to come on stage for a group photo. Please invite Professor Zhao, Dr. Poon, Professor Brown, Professor Wang, Professor Liu, Professor Wong, and Dr. Ne to come on stage for a group photo as a record, as a memory of today's session as well. Right, so let's look at our photographer. Slightly forward. Thumbs up. Classic pose. Thank you so much, ladies and gentlemen. Please take a seat. Thank you so much. We are now going to have a coffee break, so please proceed to your foyer area to enjoy and don't miss the chance to interact with other participants. And see you again in 15 minutes. 15 minutes. See you in 15 minutes time. Thank you. So welcome back, ladies and gentlemen, and we now move to session 4, cardiac disease, and we are honored to have Dr. Ne Yu, Principal Investigator and PhD Advisor at Fuwei Hospital, National Center for Cardiovascular Disease, Peking Union Medical College, and Dr. Ryo Sugimura, Assistant Professor at the School of Biomedical Sciences, the University of Hong Kong, to moderate the session. So please join me in giving them a warm welcome.
Yeah, all right, yeah, thank you very much. So first speaker, Professor Sean Wu from Stanford. His talk is Health Development and Disease Stem Cells, Engineered Tissues, and AI. Thank you very much.
All right, I've already messed up. I'm supposed to start with the introductioning about what I'm going to be talking today, but before that, to thank both Ben and Kathy for the really kind invitation to speak here in Hong Kong, which is my second time here. Also want to thank the Croucher Foundation for supporting the symposium. It's really great to see everybody here. What I'm going to talk to you about, as what I had on the slide, was basically I couldn't decide what I was going to talk to you about. So then I wrote that I was going to talk about a little bit of everything, some development, some stem cell, some engineered tissue, and some AI. And then so hopefully this will kind of tie together in some way as I work my way through the rest of the talk. So where I'm going to start is something I think many people in our field have known this for quite some time, which is the study from Eunice Frizen, where they looked at this capacity of cardiomyocyte turnover in the postnatal heart throughout the lifespan, and as you can see, that it progressively declined right from birth very dramatically until later in life. And their estimate was it's roughly about 1 to 2% turnover at around birth, and then less than 0.5% by the end of life. So that gave quite a sort of parameter around how much a normal cardiac myocyte homeostasis might be. And then you've now have heard now from many people, including for Professor Nee just now in his talk, this idea that in that very early neonatal stage, there is this transient regenerative potential for the cardiomyocyte that still has some retained proliferative capacity, so that if you do an apical amputation, as the slide here shows from Hesham Sadek's work, that you can then induce some amount of this reactivated proliferation of cardiomyocyte. And the estimate there is, again, roughly about 10% of that resection at the P1 or first stage postnatal can be slowly recover over the following few weeks. So me, as a developmental person who has been looking at heart development, we had been curious about the question of what then is this capacity for this heart within the mammalian system to be able to recover from this injury process. And then so the capacity for this myocyte regeneration in the embryonic mammalian heart. So we published a paper, and I wanted to just mention this more as a background to the rest of what I'm going to talk about, is that we did a study looking at a way to do ablation of cardiac cell in that early embryonic heart. And the way this was done is by doing a genetic system involving NKX 2.5 Cre. I think you heard a little bit about NKX 2.5, which is expressed within this early developing heart. And then another mouse, which is the diphtheria toxin mice, the Rosa 26 DTA, which is also expressing GFP cells. And then so you can see that when you do a cross of the diphtheria with the NKX, you basically take out that entire fetal heart. And so knowing this is what happened when both genes are present, what you can then do, which is what the setup that we did, is to generate first an embryonic stem cell that carry both of these, and you introduce that into the early developing blastocyst, so much in the way most people would make a genetic modified mice. And in that system, when you introduce, you make what's called a chimera, and in the chimera system, you would then get a whole ranges of different degree of ablating cardiac cells, because sometimes you might only contribute, 1 or 2%. Sometimes you may contribute 100%. And then so the way that this would work is basically if you contribute very little, then you basically get back to the same animal that you have from your breeding. But if you contribute entirely, then you basically get the same as what the knockout breeding animal would get. But what our interest, of course, is what happens if you contribute some fraction in between. So the idea then would be initially if you had, say, 50% contribution and you ablate 50% of the cell at the time when the Cree's activated, the residual cell that are not these labeled green cell with the diphtheria toxin would then presumably take over and recover. And then so when we then did the experiment, you can see that progressively as you increase the percent chimerism from the ES cells, so you can see progressively the rest of the embryo turn greener and greener, and where the heart is presumably going to be deleted, when you can get to 50 to 60% compensation, the entire embryo actually looks pretty good. Like it completely recovers in the heart ablation to look like the animals that didn't really receive much of this ablated cell. But if you go beyond that into 70, 80, 90%, then the whole embryo basically becomes significantly affected and arrested in development. So that gave us sort of the extent in which the cells are able to recover. So more than 50 to 60% of the cell losses during that fetal stages is able to be compensated. So it sort of gave us this idea that there's a pretty robust mechanism for compensating for the cell losses in that fetal heart. We then did another lineage system in addition to the NKX, which is using the alpha myosin heavy chain, and you heard some of the earlier talk using this alpha myosin heavy chain. So with alpha myosin heavy chain express Cree, it expressed slightly later than NKX, mostly in the mid-gestation, and the animal, after introducing with the same system, are largely pretty normal looking. So here with the cardiac progenitor with the NKX ablation, the heart is without any of the green cell because, as I mentioned, the compensation comes from the non-green cell, so the whole heart is non-green. But compared with the control that's entirely green, you would expect to see all green cells in the heart because it doesn't have the Cree for the ablation, right? So with the cardiomyocyte ablation, again, we also got a reasonably good recovery. So again, in that early fetal stages, both ablating at the progenitor or the cardiomyocyte stages, you're able to get pretty good recovery. And by measuring the heart size to the body weight, it's all pretty similar. And you can also measure the function. It also looks pretty similar, which then sort of raises the question, what is the mechanism which this is doing, the compensation, and what then we measure what are some of the residual cells proliferative indexes. So by measuring the Ki67 as well as measuring phosphohistone from both the cardiomyocyte ablation model as well as the cardiac progenitor ablation model, we see there was slight increase. It doesn't actually need a lot of increase in the proliferative index, and that was already sufficient to recover within about 2 to 3 days of the amount of ablation. So after we had published these papers, it started a new sort of direction of questions about, well, what exactly might be the mechanism which these residual non-ablated cell is being triggered to be able to proliferate at faster rate. And of course, many people in the field, including all of the people that have spoke earlier today in the audience, have really defined many of the proliferative pathways and processes in that developing and regenerative heart, and some of the signal that you're seeing here as well, including signal from the WIN pathway, the Hippo pathway, as well as IGF and other pathways. So, amongst all these various different signals, the one that we had studied, because one of the postdoc that joined my lab, Jan Bukima, had done a postdoc, has a PhD degree with Ibrahim Domian, where he looked at the WIN signal gain and loss of function. And in his study, he showed that if you do a gain of function study in WIN signal, you end up largely getting almost no different in the proliferation and the myocardial thickness as the control. So there really wasn't so much, but you clearly need to have the WIN signal present because the loss of function caused a significant reduced myocardial thickness. And so what was somewhat of curious to us is then what exactly is constraining the Wnt signal to induce more proliferation within this developing heart so that your gain of function doesn't give you the increases that you need. And so we then sort of wonder about something that people in the epithelial and cancer biology has known for a long time, which is this idea that when the cells are relatively sparse and have ability to proliferate, they will continue to expand and proliferate until they completely fill in the dish. And then by the time they start to completely touch each other and fill the dish, and then their proliferative activity is suppressed. So there's this idea of this contact inhibition that exists within the cells, such that when the cells are forming contact with each other, that their proliferative drive gets significantly attenuated. So that, for us, was potentially the basis for why, when you have the ablated cells and all the spaces have cleared open, that it initiates a proliferative drive that then gets filled and replaced. And so what that sort of led is a paper that we then published subsequently in looking at the ability to expand iPS-derived cardiomyocyte based on this concept. So in this paper, we described a synergism between the Wnt signal and the cell contact disruption to synergistically increase the expansion of iPS cardiomyocyte. So what we did, just a couple of slides, and this was published as well, I just want to illustrate this contact inhibition idea, is that if you do an assay with iPS cardiomyocyte, here you then set the cells up so that by day 12, you essentially treat with the Wnt signal, but either do the cells without any passaging in the same plate, or you passage them but put them back into a confined space with the exact same control, media, volume, nutrition, everything, versus you increase the plate space but maintain the same volume of nutrition and media, and then see what happens in the proliferation of these three different conditions. And what was quite interesting was that clearly the cells that have been either densely but passage versus unpassage have very similar proliferative index, just slightly increased compared with the baseline, whereas the cells that you sparsely plate so that they've always have been left to be inhibited from contacting have much more significant increase. You can see these are logarithmic fold increases in proliferation. And then we, of course, also measure that there was an increase in proliferative index. And then what became quite an interesting exercise for us is how much can you get out of this one iPS differentiation step. And then so we started this process of doing cell passaging, and you can see you can start from one small dish into many dishes and then many, many more dishes over time. So in the end, you get as much as 200 to 300 fold increase in your cardiomyocyte number, which is something that I think most people have really never seen that they can get with their cardiomyocytes. So what was interesting then when we developed this protocol, stem cell technology called us. So if you ever actually do iPS culture, people sell a lot of media. So they took a license from what the technology we have and are doing quite a lot of the selling. We don't know exactly what they're using because they won't tell us in their media, but they call it the cardiomyocyte expansion media, and is being used quite a number of people because my colleague have sometimes come up to me and say, hey, I think they're using this media recipe from what you guys have done because of what the license that they have from us on this technology. And then so what then happened with the story looking at this expansion was what exactly makes the cardiomyocyte want to go into dividing with each other when the cells are sparse. So when we look at the treatment, so here in this case, now the cells are completely on its own in these pattern surface. So iPS cardiomyocyte pattern in the 7:1 aspect ratio. If you don't treat it, it just basically fully differentiate out into these very organized sarcomere. But if you treat it with active agonist cheer, you see how there's now multiple nuclei with lots of disarray sarcomere. So but if you treat with the C59, which is the Wnt inhibitor, you can then reorganize. After dividing, the sarcomere can then remature and become reorganized again. And so this reversibility was kind of a nice feature because people sort of ask us like, well, what happened to these cells after you treat and they're all immature? Can you still do anything with it? Right? So, we sort of then thought about, well, we need to sort of demonstrate whether or not the cells can reverse and mature, and then sort of look at some of the maturation features, including the sort of cell shape, as well as their formation of cell contact, intercalated discs, as well as the sarcomere alignment. And then so what we did is create an engineered tissue using these expanded cardiomyocytes. So the assay we did was we started with these expanded versus a non-expanded cell, and then we let them fully mature over 25 more days after the expansion, and then we create these engineered tissue constructs, and then we measure both histologically as well as function that these expanded cell that then is followed by reversal does have still the ability to mature and increase the force generation over time. So that for us was kind of a nice feature, because we knew this way that you're still able to do the cardiac myocyte function after you do the expansion, that these cells are not completely sort of altered because of their persistent Wnt signal over time. So then after having published this story, we then started looking at the question of, well, then what exactly does the cell contact do to the cardiomyocyte that gives rise to this ability to, inhibit proliferation when they're in contact, but release from that inhibition when they're sort of disconnected. And then so we started looking at some of the signals. So here we looked at some of the single-cell RNA-seq and found that the sparse cells that are not in contact generally have reduced sarcomeric proteins, and also some of the disassembly within the histologic section. And we also noted that paper published from Didier Sainier in the zebrafish system, that during the process of zebrafish regeneration, there is a very active sarcomere disassembly process that in their paper was driven by an AP1 regulated signal. And in their story, they really illustrate how some of this border zone myocyte are the ones that are activated and then undergo these protrusioning process to start the regenerative mechanism, which I think is quite interesting. And there was some mammalian sort of consistency with this from Jim Martin's story with the hippo and yap. So we want to follow up a little on some of these sort of sarcomere disassembly stories. So we looked at the sparse versus the dense plated cell, and also again we saw as well that the sarcomere organization is much more reduced in the sparse cell. And then the other things that I don't have time to show is that they also increases the Wnt activity. So it actually helped enhance the Wnt signal within the cells when you're keeping the cells sparse, which is also another mechanism to increase their proliferative capacity. But one of the things that we wanted to do is figure out, are there any difference in their secretory molecule between the cells in contact versus cells that are sparse? Because we did notice that there is a density dependency to the increase in proliferation. So more cells we have in the sparse condition, the more the index of proliferation goes up. And then so we did a somewhat of a limited screen on all the secreted factor. And out of the screen, there was a few top here, including IGF-BP2, IGF-BP6, and PDGF-AA. And we then tried within the cell culture assay all three of these either separately or all together. And it turned out that the only one which has the activity is IGF-BP2, which is both in the all condition similar if you only just supplement with IGF-BP2. So this became another direction for us to look at how the IGF-BP2, which is increased in expression, both RNA and protein, in the setting of being sparse, but then is suppressed when the cells are made dense. So one of the sort of last thing that I'll mention is that when you then take the cells that are in contact, because one of the reasons why we did this is we're looking for ways to enhance that proliferation despite the fact that the cells are in contact, because, of course, you can't really do an in vivo system to disrupt contact that easily, but maybe this factor we have can help enhance the proliferation despite the presence of the cell contact. So we did an assay involving a cardiomyocyte organoid system where we generate these sphere of pure cardiomyocytes and then treat with the different conditions, including either just the base media or base media plus the cheer Wnt, or base media cheer Wnt plus IGF-BP2. And you can see that the IGF-BP2 added condition increases these protrusion-like cardiomyocyte expansions that doesn't normally happen if you just have the base B27 media. So we think that this IGF-BP does seem to provide some type of a proliferative drive that can overcome aspects of the cell contact to induce proliferation. And so that is what I have for you regarding the story around the cardiomyocyte proliferation. But I wanted to sort of go back now to sort of tell you a little bit about the story around AI work, because we have been looking at how to leverage some of the AI tool also to see whether or not you can design better experiments. One of these things is it's very complicated to design lots of experiments and then sort of end up failing because you didn't pick the right gene to do deletions and whatnot. So one of the things that we have wanted to do is to get better at the computation part. So we started this a few years ago when we got a whole lot of single-cell RNA-seq data, which is what we've been doing quite a bit, and we generated an algorithm that can predict the cell type in an automated fashion from the developing heart all the way from E6.5 to 16.5. And this algorithm basically automate the assignment of all the various different cardiac cell types during development from the single-cell RNA-seq data. And so basically, if you had a big data set, and in fact, you can even aggregate multiple studies, which is what we did, in these multiple data set aggregation, you can follow the differentiation process of cardiac cells all the way from the epiblast to the mesoderm, primitive streak, nascent mesoderm, all the way to cardiac progenitor, and eventually to the definitive cell type, cardiomyocyte, epicardiocell, endothelial cell, as well as the fibroblast. So having all these different cell type defined, you essentially create one of these computation algorithm that allows you to make prediction on what type of cells you have from the data. So we have the 90% training cells and validation and 10% of the withholds, and then we show that it gave quite a nice consistency in the prediction on its own. Of course, we wanted to use a completely separate data that's not part of the data set that we use to generate the algorithm. So we then tested a different genetic reporter system from ISLA1 Cree, which we've also have had a single-cell data set from. And then in this system, we show that the manual annotation, so these are expert developmental person annotating what kind of cells they are, matches quite well to this machine learning predictive cell type. But even down to within the cardiomyocyte, the different regions of the developing heart, the machine learning prediction also matches reasonably well. So we wanted to take this now further to see, what can you do with an algorithm like this? And that brought us recently to bring in some of the large language model analysis. So in this case, as I think many of you know, the GPT system for understanding the syntaxes, like words in books, has been tremendously powerful in being able to sort of read and understand, if not even generate some new ideas. And so we wanted to leverage in the same way of the biological data. So in this case, a transcript can be envisioned like it's a word, and then so a cell could be a paragraph's worth of words, and then a organ like a heart is a book. And then so if you take all of these transcripts in the tissues, it's essentially trying to read what is in the cell based on the genes that are expressed. And then so recently, Bo Wang's lab from Toronto has created an algorithm called SCGPT, which can combine both the RNA-seq data as well as the ATAC-seq data from single nuclei to be able to allow you to do quite a lot of various different annotation. And then so we started incorporating, and these are sort of preliminary work unpublished, that we're just now starting to look at some of the ways to take all the single-cell RNA-seq data and essentially create a gene regulatory network that are cell type specific, since we know what genes in what cells we have, and we know what gene regulatory network exists between different transcription factors. So in this case, you can create a cell type specific gene regulatory network this way, and then with that, you can then analyze the consequence of removing value from certain transcription factor within that network, and then look at the consequence of propagating the loss of that transcription factor within this computation system, and then create a prediction on what would be the consequence when a gene is deleted in this process. So we took the data set that I mentioned with all these different cells. We initiate essentially a computation run of all the transcription factor that represent major cardiac developmental genes, and then look at what the consequence of the prediction to look at the trajectory. I won't have time to go through a lot of data, but just as an illustration here, we have the various different genes that are expressed in the early developing heart, in this case, islet 1 expressed in the second heart field. We know that islet regulates second heart field, but doesn't do much to the first heart field. We apply this computation to show that if you delete islet 1, it drives the cells further towards the mature right ventricular cardiomyocyte, but it doesn't really change much of the first heart field progenitor in this case. And then it shows the loss of islet 1 really caused a major disruption in the regulatory network. Okay, so that's what I have for you today. So in summary, I have mentioned to you about the use of these sort of cardiomyocyte regenerative system to understand the ability of embryonic heart to be ablated, and that around 50-60% loss is tolerable. I show you that the cell contact mechanism is responsible for regulating proliferation, and that we can leverage this contact in the context of Wnt activation to induce massive expansion of cardiomyocytes. I mentioned also about how this contact mediated mechanism is regulated in part, at least by the secretion of IGF-BP2. I also mentioned about how the IGF-BP2 is both necessary and sufficient to expand iPS cardiomyocyte. And then finally, I show you a little bit about how some of the AI work we have in understanding developing transcriptome and how that could be leveraged to look at predicting the loss of gene expression consequence during development. Okay, and with that, I want to thank the lab folks, particularly the ones that I highlight in red, whose work I mentioned during this talk, our collaborators at Stanford, collaborators outside of Stanford, and also our funding from various agencies, and also last but not least, for all of you for your attention. I'd be happy to take any question if we have time. Thank you.
Yeah, thank you very much for the talk. Any question from audience? Oh yeah, Bean, yeah.
Hi, Sean. Very nice work. I'm wondering if in the embryonic stage, the cardiomyocytes can proliferate, and these cardiomyocytes are contact with each other in, the compaction layer. So in vivo, actually cells are contact each other from the beginning. So I'm wondering if it's the extracellular matrix that differs from embryonic to the adult stage, although they are all contact. So in addition to the cardiomyocytes, its own property, the extracellular matrix could have influence on their proliferation when they are all contacted with each other.
Yeah. So I'll probably just say that, of course, the heart cells are in contact during development the whole time. So it's not that the minute you have contact, you are automatically not able to proliferate, but it's a balance between how much drive that cells has at that moment to how much the inhibition exists within the cell. So the two kind of walk together hand in hand, but that over time, the proliferative drive really attenuates, and the cell contact is still present. And then there's a whole somewhat complicated sort of, cell sensing mechanism of what is the right number of cells that you need to have in order to maintain the proper development size. So, there are clearly other mechanisms around for sure. Aside from between the cells in the extracellular matrix, as Professor Nee just mentioned, earlier, all of it, I'm sure, plays some role in the regulation as well. But it's a very dynamic process that changes between fetal, and the postnatal and adult stages. So I don't think it's necessarily just because the cells are different or the ECM's different, but it's all kind of a combination of all the things that changes until you get to the adult stage.
Yeah, also another question is, it's good to see the Wnt signaling play an important role in the cardiomyocyte proliferation. I wonder, Wnt usually for the epithelial cells, and in the heart, for the adult heart, the ligand, the Wnt ligand, which type of cells are the major producer for these Wnt ligands?
So the adult has been kind of interesting because when people try to look at Wnt, mostly people have found more epicardial derived cell make secretion of Wnt factor, and the factor is inducing not so much a cardiomyocyte response, but more of a fibroblast response. So there's a lot of inflammation and fibrosis that are mediated from the Wnt signal in the adult heart. So I would say this kind of is making things a lot more complicated if you just try to apply the idea of the development into the adult. You're probably getting something completely different and, in fact, worse, outcome.
Okay. Thank you.
Yeah.
That was really fascinating, Sean.
Thank you.
I was interested in this effect of dense cell density on proliferation. So normally, in the normal heart development or in the normal steady state once you're a mature heart, to what degree is cell competition actually a factor in whether or not the cells will become proliferative or not?
There's been a handful of paper from Miguel Torres about this issue of cell competition. So they have generated these MYC knockout or MYC overexpress system so that they can see whether or not the ones that are induced to proliferate more with MYC is able to overcome and take over during development. And it's a somewhat complicated story, but the bottom line is that yes, there is this competition effect, so that if some cell has a proliferative advantage, they do end up become the more dominant population within the heart.
Sean, this is really amazing, and especially in the ChatGPT sort of generative AI modeling. And what you propose is that you use the gene expression profile and then eventually to build a narrative where you can predict the gene knockout phenotype. So I wonder whether you can do the reverse engineering side. And if you want to build a story of proliferation or you want to build a story of cardioprotection, can you go back and find out what's the hub genes or key genes that you need to either activate or knock out?
If I get your question correctly, you're saying that instead of destroying the normal process, can you screen to see how something might help enhance a disease-damaged heart with the same
approach? I mean, basically you have the story, right? You said, basically you have the book from the individual words, but if the model is really working, then in theory you can change any book you want.
So the closest, we haven't done that. The closest that have tried to do is from Patrick Eleanor's paper with Christina Theodoros about 2-3 years ago, where they looked at failing heart transcriptome and then implement a in silico screen approach to look to see between the healthy transcriptome and the failing transcriptome. If you then implement this computational simulation, are there any gene loss you find that reverts that transcriptome closer to the healthy? So that I think is probably the closest in terms of what you're saying about going from disease to normal. Development is a little different. I think it's hard to sort of go from a disease develop and then go back to normal develop. I mean, I guess you could, but it's probably harder than if you just had a failing phenotype and revert to a healthy phenotype.
So thank you very much for a very interesting talk. So your Wnt signaling reduced cell contact protocol. So I noticed that you applied a protocol to cells when they're still very proliferative, like day 11. So at which stage of maturation do the cells stop responding, or do they keep on responding when they progressively gain in maturation?
So we really had wanted, when we first found this phenomena, to know, can we reverse that fully mature cell to become proliferative again? And that, we were pretty excited to try. When we try to do this in the cells that's now day 60, when they're fully no longer proliferate so much, unfortunately, when you then apply the Wnt signal, they just don't respond nearly as well. So I think most of what you see and how you get a lot of cell happens much earlier is because you need to keep them immature, and as much as you can keep them immature so that they don't become more mature. Because one of the things people have asked us after we published the paper, and they have tried the same, they say, the protocol really works, but I never get as much cell number as you get. Like, we'll get about 50-fold, 70-fold, but we never quite do a 200, 300-fold. And usually the reason why they're not getting as much is they're not splitting it as often. So they let the cells touch and then they split. And I think every time when the cells are touching, you're losing some of that capacity. So when they split more often, they can usually stretch the whole process for far longer.
So do you know what is blocking, shall we say, blocking the response of the cells to respond? So so you said that so later stage cells, they don't work anymore. So that's because of prior cell contact, or is there any kind of maturation block that stops them from responding to the Wnt signaling?
Yeah, I think there is a full-on maturationing that happens so that they're accelerating the maturation far faster when they're staying contacted to each other, because a lot of other maturing parameters, sarcomeric organization, contractile force, all that goes along with when they're in contact with each other. So I think, those things are very hard to turn back, once they've already been set in place, unfortunately.
Thank you so much.
Yeah.
Okay, thank you, Professor Xin Wu. Again, thank you. It's my honor to introduce next speaker is Professor Hui Zhao. Hui Zhao is a professor Chinese University of Hong Kong. His research focuses on early embryonic partnering, genomic editing, and cardiac development regeneration. And he has published over 114 papers in leading journals, including Science One's Nature Communication, PNAS Development, and the EMBO Journal. His research is supported by major funding agencies, such as the Ministry of Science and Technology and the National Natural Science Foundation of China. Let's welcome Professor Zhao. Thank you.
Thank you so much for your introduction. I also thank Professor Cassie Liu and Professor Ping Zhou for invitation. I'm a developmental biologist. I'm using frog and zebrafish model system. So actually, I'm the novice in this field. Actually, I think that's a great opportunity for me to learn from senior investigators in this field. I learned a lot. Yeah, I guess everybody understands the significance to study the myocardial infarction, and we learned a lot from Professor Nie Yu and Professor Wu. So I guess it's actually a global burden, and current therapeutic approach is not sufficient. And if we can regenerate our heart, so regeneration jeopardize our heart tissue by promoting the cardiomyocyte proliferation, migration, and reconstitution. That would be great as a revolutionary therapeutic approach. However, our heart is the least organ for regeneration. And in some lower animals, if we go down back to some lower animals, for example, zebrafish or the frog larvae, their heart can be fully regenerated. So if we perform the apic resection or cryo injury to the zebrafish heart, after injury, the zebrafish cardiomyocytes start proliferate day 4 after the injury, and then reach the peak on day 7. So that's kind of increased cardiomyocyte proliferating, maintains stably, and then decline, go back to the normal level, day 15. So we have the model. Okay, we have the zebrafish model and frog model and mouse model in our lab. These are representative animal models, or representative the evolutionary distinct vertebrate, and have the progressively increasing heart complexity. For example, the zebrafish heart contains only 2 chambers, and frog heart contains 3 chambers, and mouse heart, of course, everybody knows the 4 chambers. So if we do a cross-species analysis, perhaps we can identify some mechanism that has been silenced during the evolution. We decode the silenced mechanism, and coming back, perhaps can be used for the therapeutic approach. And I have been using frog as a model system even when I was a PhD student. So I have been thinking for the tissue regeneration. I have been thinking for many years the thyroid hormone perhaps detrimental for the tissue regeneration, because I have some evidence. If you look at the frog larvae, some organs in frog larvae, they can be regenerated. For example, the lens, as well as the limb, they can be regenerated when they were a larvae. However, when it's approached to the metamorphosis, so that's kind of capacity decline and eventually disappear. So if you go back to the, if you look at the lifespan of the frog, frogs after the fertilization, they become gastrocneural and then become larvae, and then they undergo a so-called metamorphosis, so become froglet, become frog. And in the larvae stage, organs can be regenerated, and become the froglet, the regeneration capacity disappears. So what happens? So certainly a very important period, metamorphosis before and after. The regeneration capacity, you see the big difference. And so what's happening in the metamorphosis? I guess everybody knows the thyroid hormone play a very, very important role for metamorphosis. It's a kind of driving force for metamorphosis. And indeed, we look at qPCR to visualize the thyroid hormone receptor, and we can see the surge of the thyroid hormone receptor alpha, thyroid hormone receptor beta expression during the metamorphosis. So for the heart, indeed, the frog tadpole heart can be fully regenerated. However, again, like other organs, this regenerative capacity disappears during the metamorphosis. And indeed, study from a friend group suggests the explorer thyroid hormone or thyroid hormone depletion will actually interfere the heart regeneration. And also another interesting publication from Professor Bohan's lab Bohan is in UCSF, and he found when the thyroid hormone signaling was blocked in mice, the heart show a significant regenerative capacity after injury. And they also performed the cross-species analysis. So they suggest that the species with lower metabolic rate, body temperature, and the thyroid hormone level maintain more diploid cardiomyocyte. And diploid cardiomyocyte, certainly everybody knows that's the very important parameter to assess the heart regeneration. So diploid cardiomyocyte advances inversely correlated with the standard metabolic rate, body temperature, and thyroxine level. And so thyroid hormone, we know thyroid hormone even we were in primary school. And thyroid hormone is produced by thyroid gland at precursor. There's thyroxine, the T4, and T4 will be converted in T3. That is triiodothyronine. That is the active form. And the thyroid hormone require two types of receptors, retinol X receptor and a thyroid hormone receptor. Without the thyroid hormone, the RXR and thyroid hormone receptor, they form a protein complex and then bind to a co-suppressor. So this is a kind of protein complex suppress gene expression. And after binding to the thyroid hormone, this binding will cause conformation change of the thyroid hormone receptor. So the co-suppressor will be replaced by co-activator. So the entire protein complex will become the gene activator. Certainly, you can imagine after activation, they will change the gene expression landscape. I mean, landscape, indeed, a landscape, not single gene, but a large group of genes. And so we thought we can do something perhaps with thyroid hormone and its receptor. We downloaded single cell data from published paper from the public database, and we found the thyroid hormone alpha. One is expressed in various cell populations, mouse heart, including cardiomyocyte and fibroblast and smooth muscle cells. And we also collaborate with Professor Xuan Zhang from NIH. She generates the serial hormone Alpha-1 zero-free mutant. By the way, so zero-free genome has partially duplicated. So the serial hormone Alpha-1 has two paralog in zero-free genome. So THRAA and THRAB. So THRAA is the major form in zebra-free heart. So we obtained the mutant line from Professor Xuan Zhang. So we get the THRAA mutant. This mutant contains 8BP insertion at THRA locus. So heterozygous of the mutant looks very normal. Homozygous mutant show a heart developmental defect, and it was described as a dislocated atrium, and that's very interesting. And I will, if I have a chance, I will talk to this in details. And this, this mutant line contains a 18 insertion BP at the THRA locus, leading to an early stop of the serial hormone receptor. And this truncated protein cannot bind the serial hormone anymore. And we performed the apex resection as other people did. So we performed apex resection and evaluate the cardiomyocyte proliferation and evaluate the fibrosis area. So we found enhanced cardiomyocyte proliferation on D7 and D15, and we also observed reduced fibrotic area in THRA heterozygous mutant heart. And we spent a long time to establish the ECG measurement platform, so we can do some ECG measurement to assess heart function. So in some of the parameters, ECG parameters, for example, the TP duration, we find the improved functional recovery. And next, we want to identify the underlying mechanism. So what we learned from the our preliminary data, so the knockdown or partially loss of the serial hormone receptor THRA were beneficial for the heart regeneration. So we want to study the underlying mechanism. So again, we perform the apex resection and then collect injury zone at a different time point and do a background-seq. At that time, we haven't managed to perform the single nucleus sequencing. So we go ahead with the background-seq, and certainly we can see after we obtain the background-seq, we have many comparisons. So we can compare the gene expression profile between the different time points versus uninjured stage, and certainly we can compare the gene expression profile between the THRA mutant versus wild-type mutant. So I show some what I learned, some of the from the bioinformatics analysis. So what I really learned is loss of the serial hormone receptor does affect inflammatory gene expression. So at least a large group genes related inflammation is reached the peak earlier than the wild type. And we also find the more neutral field related genes are peaked at 8 hours post injury. And we found the M2 macrophage related genes are used at D7 in THRA mutant heart. And we also evaluate the predict the genes expression that regulates cell proliferation. So we found the cardiomyocyte proliferating window extend to extending to D7, and in THRA mutant, well, the wild type zero-phase heart at that time, at the injury zone, the cardiomyocyte already undergo differentiation. Differentiation is a term we got from the Go term, but in fact, I think structural maturation perhaps is the best way to describe this stage. And we look at the DEG, analyze the DEG promoter. So we found the inferred transfer factors in the upregulated DEG at D7 favor to the cell proliferation. Regarding the metabolic pathways, so we found the glucolysis is slightly deviate in the THRA mutant. However, we find a clear difference in dynamics in the pentose phosphorate pathway, as well as the mitochondrial signal pathway and the macrophage signal pathway. And we performed the single nuclear RNA-seq for the injury zone, and we can identify 13 different cell population in the injury zone, and we found the THRA is enriched in the cardiomyocytes, in the fibroblast and other cell type, neuronal cell type, and we also develop a method that can predict to identify the cell population regulated by the thyroid hormone receptor. So we found the cardiomyocyte and the adipocyte and neurocyte perhaps is most affected cell population in heart by the thyroid hormone receptor. And in the cardiomyocyte, we found the TNF signaling and the oxidative phosphorylation signaling pathway are most affected by the thyroid hormone receptor. And so we next narrow down our focus. We focus on the hypoxia signaling pathway, and in particular on the HIF3 alpha, this gene, because we found the this HIF3 alpha genes strong upregulation of the HIF3 alpha observed in THRA mutant after the later reproductive stage. And by analyzing the ChIP-seq data, we predict that the thyroid hormone alpha-1 can directly bind to the HIF3 alpha promoter. And from the RNA-seq and our own qPCR result, we found thyroid hormone receptor alpha can regulate HIF3 alpha expression. So we have been it has been well established that HIF signaling pathway is play a very important role or beneficial role for the heart regeneration, and there are numerous studies have been published. And HIF1 alpha and the or HIF3 alpha combine to HIF1 beta to form a complex enter to the nucleus to activate downstream gene expression. And HIF3 alpha is kind of different from the HIF1 alpha and HIF2 alpha because it only contains one TAD domain, the NTAD domain. So conventionally, people believe, HIF3 alpha it perhaps is a competitor to the HIF1 alpha or HIF2 alpha, negatively regulate HIF signal pathway. On the other hand, there are many spreading variants from the HIF3 alpha, and increasing studies suggest that HIF3 alpha has a HIF-independent function. So we use CRISPR-Cas9 to knock out, to disrupt HIF3 alpha genes, and we actually carefully remove the region that code NTD domain. That the mutant has been confirmed by genotyping and confirmed by RT-PCR, followed by the sequencing. The heterozygous HIF3 mutant line and developed quite normal, and there's no evident defect. However, if you look at the HIF3 alpha homozygous mutant, and I would say many embryo died during the development, but some of the embryo they can manage, grow up, become adult. So if you look at the adult HIF3 homozygous mutant fish heart, we find a enlarged atrium very clear in the HIF3 alpha homozygous mutant. And we carefully visualize the atrium and ventricle. And indeed, we found the enlargement of the atrium, and there are a lot of blood cells accumulated in the atrium. And so that's the phenotype we get. And we go back and use human cytohybridization, look at the embryo of the HIF3 alpha homozygous mutant, and we found the heart looping is affected by loss of the HIF3 alpha. And we also established the ultrasound, the echocardiology for zebrafish. So we found we measured the function of the zebrafish heart. So we found a peak of velocity in the HIF3 alpha homozygous mutant is significantly high, indicating an abnormal increase of the heart flow crossing the valve into the ventricle. And again, we go back. We use heterozygous HIF3 alpha mutant to perform the epic resection. So we found a impaired heart regeneration because there are larger fibrillary area in the HIF3 alpha mutant heart compared to the wild type. And also you perform ECG, and some of the ECG parameters reveal that the greater degree of the, for example, greater degree of decrease of the TP durations suggests the impaired functional recovery. And so that's because of I think that's because of the persistent scarring. And I guess I can stop here. I want to make a short summary. So we found that a moderate reduction of the thyroid hormone and thyroid hormone receptor alpha signaling promote heart regeneration in zebrafish. We show some evidence, and we found HIF3 alpha is regulated by THRA in zebrafish. So disruption of the HIF3 alpha impair heart function, and homozygous HIF3 alpha mutant zebrafish show reduced heart regeneration capacity. And so I want to thank my collaborator, Professor Xuan Zhang from NIH and Professor Ge Wei from University of Macau. And this work has been done by 3 students. Now they are graduates. Dr. Jiang Chunwei, Dr. Man Yi Zhang performed the bioinformatics analysis. Dr. Imatis Hassan performed the ECG and ultrasound cardiology analysis for the heart. I also thank my funding from the RGC, from ITF, and from Provotos Life Sciences. Thank you so much for your listening. Thank you very much.
Thank you very much for Professor. Any questions? Do you have any question? I have a small.
Thanks for Professor George exciting sharing. So I know that the thyroid hormone will bind to the RXR, which is a retinoic acid receptor. So and I wonder whether this T3 is play a competitive role with RA, retinoic acid.
That's a good question. Although I actually know something about the retinoic acid signaling pathway, but I know nothing about retinoic acid and thyroid hormone signaling. I don't know that often.
Oh, thank you, Professor.
Any question? I have a question. We know that T3 is related to the body temperature.
Right.
Yeah. I want to know whether you moderate the T3 receptor and the zebrafish, the body temperature measure could be changed.
To be honest, we haven't tried to measure the zebrafish body temperature because
it's hard to detect it.
Yeah, I don't know how to find a right place.
Maybe we can culture it in cold water.
Yeah, fish needs to be cold water.
Okay. Thank you. Thank you, Professor Zhao. Again, thank you.
Yeah, thank you very much. So next speaker is Professor Li Zhang from Shanghai Jiaotong. Talk title is NRF3 in coronary heart disease continuum, novel therapeutic approach network. Thank you very much.
Thanks for kind introduction. And I sincerely thank Professor Kathy Liu for her kind invitation. And it's my great pleasure to be here in Hong Kong to present our recent work about NRF3 in cardiovascular disease and talk about the potential as a new therapeutic target. So as we know, cardiovascular disease is still the leading cause of death, and the coronary heart disease is one of the main contributors. So importantly, we should not we should not look at coronary heart disease as a single event. It represents disease continuum, including vascular pathology, thrombosis formation, and myocardial injury. So these stages are closely linked and together driven the progress and clinical outcome. Targeting key events across this continuum remain a big challenge and opportunity. To explore this question, so we focus on nerve family of transcription factor. So including NRF1, NRF2, and NRF3 belong to Kaplan-Coller or CNC family. These proteins are involved in regulate oxidative stress, metabolism, and cellular homeostasis, all of which are highly relevant to cardiovascular disease. So among the nerve family member, NRF2 is by far the most extensive studied. It is well known as the central regulator of oxidative stress, as has been shown to play protective roles in multiple cardiovascular cell types. Because oxidative stress is a common driver of cardiovascular injury, so NRF2 has attracted considerable interest as a potential therapeutic target. However, what makes NRF2 so important also makes it difficult to target therapeutically. So NRF2 is broadly expressed, which limits disease specificity and raises concern about off-target effect. In addition, its function is highly contact dependent. In some settings, NRF2 activation is protective, but in other, especially when dysregulated, it may also contribute to disease progress. So we think about, is there any more selective and potentially safer target may exist within the same families. This is why we focus on NRF3. Unlike NRF2, which is broad expressed and regulates general cellular homeostasis, NRF3 is expressed at a very low level in most normal tissues under physiological conditions, but under pathological conditions, its expression becomes markedly increased. So we observe elevated NRF3 expression in human atherosclerotic formal arteries and in myocardial infarction tissues. So in our recent published work, we show that inhibition of NRF3 attenuates small muscle cell phenotypic switching and through TRIM5 mediate autophagy and reduce stent-induced restenosis. Importantly, this was also validated in a porcine restenosis model supporting its translational relevance. So we then extended this work to myocardial injury. In a separate work, we found that NERV3 promotes injury-induced cardiac remodeling and impair cardiac functions by accelerating mitochondrial superoxide and cardiomyocyte apoptosis. So at this stage, NERV3 appear to be involved in both vascular remodeling and myocardial injury. So this study has also attracted encouraged attention from other cardiovascular researchers. So Professor Stellers highlights the possibility that NERV3 may drive disease-related phenotypes, specifically under pathological stimulation, suggests a more selective role compared with NERV1 or NERV2. This further strengthens our view that NERV3 may represent a more specific and potential safer therapeutic target in cardiovascular disease. So while studying myocardial infarction, we collect not only human cardiac tissue, but also peripheral blood samples from the same patients. So unexpectedly, we found increased NERV3 expression in patients from acute MI patient. So this was particularly interesting because thrombosis is the key event linking vascular pathology to acute ischemic injury. So we asked whether NERV3 may also regulate platelet function and thrombosis. So we first analyzed public available platelet transcriptometry data sets. We found in the condition associated with high thrombotic risk, including sepsis, SLE, and acute MI, platelet NERV3 expression was markedly increased. This suggests that elevated platelet NERV3 may also associate with thrombotic state in humans. So we then examined this question in our own clinical sample. Consistent with the public data sets, NERV3 expression in platelets was a significant increase in patients with acute MI and compared with both healthy control and patient with stable angina. So we then, more importantly, this increase was positively associated with platelet hyperreactivity. This suggests NERV3 is not only a passive marker, but may be functional linked to thrombotic risk. To test this hypothesis, so we generate platelet-specific NERV3 knockout mouse. So use a ferric chloride-induced carotid artery injury model. We found that platelet-specific loss of NERV3 led to reduced thrombosis formation and prolonged occlusion time. This data indicated platelet NERV3 promotes arterial thrombosis in vivo. We then asked whether this effect could be generalized across other thrombosis models. Indeed, similar results we observed in both mesenteric arterial thrombosis and pulmonary embolism model. This consistency suggests that the thrombotic role of NERV3 is robust rather than model specific. We then extend this analysis to organ microthrombosis in species using the CRP model. In this setting, platelet-specific NERV3 deficiency reduce organ microthrombosis and improve sepsis-associated cardiac dysfunction. So platelet NERV3 appear to contribute not only to large cell vessel thrombosis, but also to microvascular injury. So when next examine platelet function in vitro, NERV3 deficient platelets show impair aggregation, reduced spreading, and defective clot retraction in response to multiple agonists. As we know, platelet activity is a highly energy-intensive process. Glycolysis and mitochondrial oxidative phosphorylation are engaged to rapidly generate ATP to meet high energy demand during the activation. Since NERV3 has been linked to metabolic regulation in other contexts, we hypothesize it might affect platelet metabolism. NERV3 deficient reduced total ATP contents in the sepsis setting, but had little effect on the shame conduction. By contrast, it did not significantly alter glucose uptake in other resting or activated platelets. So this suggests that NERV3 does not primarily regulate glucose entry, but rather affect ATP generation downstream. So the metabolic analysis shows that glucolytic pathway was significantly altered in NERV3 deficient platelets. Glycolytic stress test further confirms that loss of NERV3 impair glycolytic function. So this suggests that NERV3 plays an important role in maintaining platelet and glycolysis. So we next look more closely at individual glycolytic intermediates. Interesting, 3-phosphoglycerates or 3-PG was significantly reduced in NERV3 deficient platelets, where all upstream metabolites were largely unchanged. So this points us towards phosphoglycerate kinase 1 or PGK1 as a potential downstream mediator of NERV3 function. So NERV3 is classically known as a transcriptional factor, but platelets are nucleate cells. So NERV3 is unlikely to function through a classical transcriptional regulation in this context. So we found that NERV3 knockout had no obvious effect on PGK1 expression, where we do find that the interaction between NERV3 and the PGK1 increased during platelet activation. So molecular docking support this interaction, and Cherkasshin analysis suggests that the 5312694 amino acids region of NERV3 are important for PGK1 binding. NERV3 deficiency significantly reduced PGK1-mediated 3-PG generation and partially decreased ATP production. So in addition, PGK1 inhibition impaired platelet activation in control group, but had a minimal additional effects in NERV3 deficient platelets. So for the in vivo thrombosis experiments, we use a platelet transfusion-based trimeric model. So water type recipient mouse receiving platelets from water type or NERV3 deficient donors. So this platelet was pre-treated in vitro with PGK1 with or without CBR. It's inhibits before transfusion. In mouse receiving water type platelets, so PGK1 enhanced thrombosis formation in both colonnade artery and mesenteric artery model, and this effect was attenuated by CBR. By comparison, the PGK1 associated thrombotic response was have no effect on the NERV3 knockout group. So as there are currently no conventional NERV3 inhibitors, so we perform virtual screen and identify K-hardening A or Ka as a candidate compound with high predicted affinity for NERV3. So docking analysis supports that Ka binds within the NERV3 PGK1 interaction region. So functionally, Ka disrupted NERV3 PGK1 binding after thrombosis stimulation, and SRP confirmed stable binding between Ka and NERV3. So we next evaluate the function effect of Ka on platelet activation and thrombosis. So Ka suppressed the key platelet activation-related response, including platelet spreading and shear-induced thrombosis. So in our ischemia perfusion injury model, Ka treatment also reduced microthrombosis formation in the heart. So this finding provide proof of conception that targeting NERV3 may offer a new therapeutic strategy for reduced thrombotic injury in cardiovascular disease. So this study shows In summary, NERV3 expression is elevated in patient in the platelets from the patient with acute MI and associated with platelets hyperreactivity. Increase NERV3 facilitated platelet activation and thrombosis by interaction with PGK1 and promote its glycolytic activity. So therapeutic strategy targeting NERV3 PGK1 interaction may offer a new and effective approach to anti-thrombotic treatment. So to conclude, NERV3 was initially reported in the context of cholesterol metabolism, but our sequential work has progressively expanded its role across multiple forms of cardiovascular disease, including neurofilament hyperplasia, myocardial infarction, and more recently thrombosis. And some ongoing work also focused on atherosclerosis and cardiac remodeling. So this work was the platelet-related work was carried out mainly by Mr. Zhou he during his PhD study. And I would like to thank Professor Zhou Bing and Professor Qin Bosch and Professor Qin Zhongxiao for their valued support and thank the support from the government funding. That's all. Many thanks for your attention.
From audience? Yeah, we'll be starting. So this KA drug could be potentially interesting for like prevention of atherosclerosis. So have you tried this one in animal model to see its different prevention power?
Thank you. As I mentioned, we just virtual sequencing the KA and to test some effect on thrombotic condition, but we have not tried the other pathological pathological model in animal. Yeah, thank you.
Yeah, okay. Any question from audience? Yeah, for the sake of time, yeah, we move on to our next speaker. Yeah, thank you very much.
The next speaker is Professor Kathy Chen. Professor Kathy is the Emeritus Chair of Biochemistry and Professor at the School of Biomedical Science, the University of Hong Kong. Okay. She has published research articles in leading journals such as Nature Genetics, eLife, PNAS, and JCI. She is senior editor for the Journal of eLife. Okay, she is the vice chair and the co-chair of the GRC and developmental biology for 2025 and 2027 respectively. Let's welcome Professor Kathy. Okay.
I'd like to say thank you very much to Kathy and VII for the kind invitation to take part in this very exciting symposium. And I think for many people here who know me, they find it rather surprising that I'm talking in a cardiac symposium. But I hope it will become obvious why I'm talking in such a cardiac conference. And the main theme of today's talk is to illustrate the importance of the extracellular matrix. I think we've had several people mention the extracellular matrix, but I'd like to share with you some insights that we have gained through studying the consequences of loss of function of a key extracellular matrix protein in regulating cardiac development. So a lot of people think the ECM, the extracellular matrix, is boring. It's a structure, it's sort of stuff that's surrounding cells. And I'd like to, hopefully at the end of today's talk, you will begin to appreciate that the extracellular matrix is not just there for structural support, but it actually has an important role in modulating the availability of morphogens and growth factors. And I'm going to illustrate that by talking about one particular extracellular matrix protein, procollagen 2A, and its expression in the heart. What happens if you knock out 2A, and how we discovered through knocking out 2A that the 2A procollagen has an important role as a mediator, not of growth factor availability, and thereby also the transcriptional activity for the proper development of the heart. The work actually started a very long time ago when I started to do expression analysis. And collagen 2A is well known to be the major component of cartilage. It was thought, and still in some papers, collagen 2 is described as cartilage specific. But a long time ago, when I actually went to London to learn how to do in situ hybridization in my friend's lab, Robin Lovell-Badge, I found out that collagen 2 was not only expressed in cartilage, but was expressed in early development in the mouse embryo. It was expressed in many different places, and surprisingly and relevant to today's talk, it's very strongly expressed early in mouse development in the heart, the developing heart. And also we had a study where we also found that it was very strongly expressed in early human embryos. So this is in situ hybridization of a day 30 human embryo. So this was very interesting, and so we published a number of papers describing the distribution of type 2 collagen expression in different tissues during mouse development at different stages. But then we also discovered that actually collagen 2, which is normally synthesized as a procollagen, which is a triple helical domain, and it has two globular domains, the C-propeptide. And we discovered, together with Linda Sandell, that the COL2A1 gene has got an alternative exon, exon 2, which encodes part of the N-propeptide, and this extra alternatively spliced exon encodes a cysteine-rich domain, which is very similar to what you find in some of the proteins like caudin, was an example. And so we embarked on expression analysis and found that actually the 2A form of procollagen was specifically expressed early in development in a number of different non-chondrogenic tissues. It's also expressed in cartilage, but mainly expressed early in development. And so we published a number of papers showing the specific and differential expression of the 2A form of procollagen in both human and in mouse. So it was very interesting, and a number of students actually contributed to this, and this is here, and this was a major collaboration with Professor Patrick Tam, who helped at that time. I was just learning mouse embryology and learning to identify all these different cell types in tissues in mouse development. So the interesting thing, as is illustrated here, is that the 2A form is expressed very early in the heart, in the cardiac crescent. And as you can see, it sort of abuts onto the expressing region for islet 1 and overlaps with the expression of NKX 2.5. So these are two very important transcription factors for heart development. So this is a hallmark, it's very early in the mouse. Now, procollagen 2A was also reported by Linda Sandell's group, by EMs and immuno-EMs, that this actually existed as fibrils, and these N-propeptides, this is an antibody, immunogold antibody specific to the N-propeptide, sort of sticks out. So if you can imagine, in the early mouse embryo, actually there's very little extracellular matrix at that stage. Most cells are very densely packed. but there are also these collagen fibrils with these N-propeptides sticking out, and they're not actually removed. So this is the selection of tissues in which we could find expression of 2A procollagen in human embryos. And we looked at stages 27 to 44 days, and very importantly, relevant to today's symposium, the expression of 2A in the epicardium and the myocardium of the developing heart. So what regulates this gene in this particular fashion throughout? So in collaboration with Miranda Ramilson in Sydney, she had developed a method to predict enhancers in human enhancers genome-wide, and to look for enhancers in the COL2A1 locus, and to see whether there were any potential enhancers that might be related to the heart. And as you can see from their analysis, there was actually definitely some peaks that were associated with the heart in the COL2A1 locus. So that implies that there were a number of regulatory elements that was driving COL2A1 expression to the developing heart. And when we looked to see whether there was any region that was particularly conserved across many species, we identified a block that was upstream, this thing we call block C, which was upstream about 4.5 kb upstream of COL2A1, which was conserved across a number of mammalian species. So when we looked very carefully at this region, very interestingly, we found the binding motifs that were characteristic of binding motifs for a number of transcription factors which are important for heart development, like SRF, GATA4, MEF, and KX2.5. So that suggested that this conserved region might be an enhancer element. And so in a number of transgenic reporter experiments, as well as mutating it, we tested out the ability of this region to drive expression to the developing heart, and we harvested these transgenic embryos at E9.5 days, in 9.5 days. And we used a BAC which contained the entire collagen 2 gene, including the this region, which is the enhancer. And so you can see that this BAC gave you expression in like Z staining specifically in the heart. And then if we disrupted this region here, which we knew contained the NKX2.5, GATA4, and everything that we identified, we actually lost specifically the heart expression. But you can see that the paraxomesoderm expression is still retained. So then we started to mutate each individual binding site for the different transcription factor, NKX2.5, GATA4, MEF2C, SRF, and then combinations of them, and all three. And it seems that if you individually mutate those binding sites, you get partial loss of cardiac expression. But if you mutate all three, NKX2.5, GATA4, and MEF2C, then you completely lose cardiac expression. So I think this was evidence that this region that we had identified was a cardiac-specific enhancer. So then, what is the significance of the expression of this isoform of procollagen 2 in heart development? So we knocked out the 2A form specifically, leaving the 2B form. But I also forgot to say that in early heart development, up to about 12.5 days, the only form of procollagen 2 that's expressed is the 2A form and not the 2B form, the one that doesn't have the extra domain. And so what we found was, in fact, there were significant cardiac defects in these knockout mice. And indeed, so this is a histology of a normal heart, developing heart at E9.5 days, and also at P1, sorry. And you can see that there are major defects Here, for example, there's a hole in the septum and another bigger hole in here, and there was a problem with the heart looping. And at the early stage, at 10.5 days, you can see significant deficiency in the endocardium of these 2A mice. So it leads to, and this is a summary of all the defects that we scored at birth for these mice. And you can see some of these, I mean, for those of you who are experts in heart development, you recognize these terms: DORV, double outlet right ventricle, transposition of the great vessels, persistent truncus arteriosus, and so forth. And all of these defects are characteristics of congenital heart disease, which is traced to a defect in the outflow tract and also the second heart field. So that indicates that the 2A form was very important for the proper development of the heart, and if you disrupted it, you would get a phenotype that was typical of congenital heart defects that were due to an outflow tract problem. And so just a little bit of background for those students who are not familiar with the development of the heart. We can see that actually very early on, at the cardiac crescent stage, you begin to have two regions. One is the first heart field and then the second heart field. And this in color gives you the contribution of the first heart field and the second heart field to different compartments of the heart. So you can see the outflow tract, which is in green, that comes from the second heart field. First heart field is in pink. And if you cut a section through there, you will see that the second heart field lies very closely, very close to each other in the splenic mesoderm. And so, and this region here contributes to the heart tube and also the second heart field and to the dorsal mesocardium, but not to the pericardium. And so this is more or less, again, another representation of the contributions of the first and second heart field to the final fully formed hearts at 15.5 days. And you can see they have their different contributions. So if we then look at all of these defects, and bearing in mind what we found in terms of the binding of all these transcription factors to this enhancer, and we know that all of these, remember I mentioned NKX 2.5, GATA4, ILAT1, they were all important, identified as key cardiac transcription factors. Also important in the heart development, this early stages, was TGF-beta signaling, Notch signaling, and retinoic acid, for example. And if you have a disruption in tetralogy of Fallot, for example, if you mutate NKX 2.5, TBX1 in FOG, you have some of these phenotypes which we found in our mice, the tetralogy of Fallot, double outlet right ventricle. These are all associated with mutations in these transcription factors. So we ask the question, what's the relationship of 2A, an extracellular matrix protein? How was it related to these transcription factors? It's possible these transcription factors actually regulate COL2, but how does it actually also, does it have any effect on these transcription factors? And to our surprise, we actually found that, for example, ILAT1, which I showed you was really important for the second heart field, in the 2A mutant, ILAT1 was drastically downregulated. NKX 2.5 was also downregulated. And indeed, in a later stage, it's at E8, at 9.5 days, the outflow tract and the endocardium has hardly any NKX 2.5 expression. And COL2A1 itself was also downregulated. So this is raising an interesting concept that the transcription factors that could be regulating COL2 and COL2 itself were all being affected in this 2A mutant. And what might be the significance of that? And so I collaborated with Richard Harvey and his postdoc Owen Prell, who had generated NKX 2.5 mutants. And so we looked in the mutants to see what happened to COL2A1. And we could see that COL2A1 is downregulated, and this is the in situ hybridization that shows that, was downregulated in the NKX2.5 mutants. And then in collaboration with Tim Moen, we also and his postdoc, Catherine Shao, we actually found that using that particular different regions of that enhancer region, we found by chip PCR that NKX2.5 bound significantly to this region C, which was the enhancer that we had been testing out in the transgenic analysis. And then in collaboration with William Poole, we looked at the GATA4 mutants, and indeed we also found that the GATA4 mutants also had downregulation of COL2A1. So that suggests that COL2A1 and NKX2.5, together with the enhancer assays, was regulated by these transcription factors. And further, when we did GATA4 chip, we could find again for that same region C, which bound NKX2.5 bound to GATA4 also bound to that. So these data suggest that this region, this enhancer, was directly regulated by these key transcription factors. And so so then the question is, what is the underlying mechanism behind the heart defects apart from, and why are these transcription factors themselves downregulated in these mutants? And so we began to look to see what might be, whether growth factors, because the extracellular matrix binds to growth factors, so was it important in making, modulating the availability of growth factors? And we looked to see that indeed there were a number of growth factors, BMP2, 4, TGF-beta1, which are important for early heart development. And they are also, and so these are all the transcription factors that I've been mentioning so far. So what about type 2A then? Is type 2A a regulator of BMP and TGF-beta signaling? And there had been some previous work done by Linda Sandel's lab, which showed that 2A procollagen in cartilage actually binds to TGF-beta1 and BMP2. And Eddie Robertis' group had also shown that if you ectopically express 2A procollagen in Xenopus embryos, it had a dorsalizing effect, and that it bound BMP4. And the dorsalizing activity is indicative of an antagonistic effect, meaning 2A would be inhibiting BMP activity. But what about in our loss of function? That was in a gain of function experiment. What about in our loss of function experiment where we knocked out 2A? What is the function what is the impact of losing 2A? And this actually shows you the expression pattern of BMP2 compared to 2A. And you can see there's very, very close relationship between the expression of 2A and these BMP2 and 4. And so when we looked as the readout of BMP signaling, which is phosphorylated PSMAD158, we found that there was in the outflow tract a significant reduction in signal for PSMAD158, and indeed in the 2A null mice there's practically some loss of it in the outflow tract. And at a later stage, and this defect in signaling persisted so that at E13.5 you can see there's very little nuclear staining for PSMAD158 in the 2A heart. And we further then did so SMAD4 is a downstream facilitator of the BMP signaling, and so we looked for genetic interaction between 2A and SMAD4 by making genetic crosses, and we can see that in compound heterozygotes, the frequency in compound heterozygotes of abnormal heart looping was significantly elevated compared to the heterozygous mutants for both SMAD4 and wild type as well. So finally, is 2A a facilitator or an antagonist? So we set about, it was quite difficult to try to do this experiment, but in the end we decided to try out using a reporter a luciferase reporter that reports TGF-beta BMP signaling activity. And so we then co-expressed transfected different constructs to test on their impact on enhancing activity of the reporter. The empty vector, so that's the control. If you add 2A, you have a significant increase in signaling activity. If you add 2B, which is the one without the alternative region, you didn't get anything. Interestingly, when you add 2A plus 2B, you basically didn't get anything either. And if you added the N-propeptide by itself, you didn't. So this was a very interesting experiment because it told us you needed the full-length 2A procollagen. You cannot have a hybrid 2A, 2B procollagen because it would not be functional. And the N-propeptide alone is not sufficient. So you need the entire procollagen molecule for that. So I think based on all of these results, we think that the 2A procollagen is a facilitator of TGF-beta BMP signaling. And so now this is our model of procollagen 2A, and it's a mediator of a positive feedback loop that controls the cardiogenic transcriptional network and actually which also controls itself. So you have regulation in the cardiomyocyte of the COL2 by these NK, all these transcription factors, which regulate the expression of COL2A1. COL2A1 gives you 2A protein, which binds to BMP TGF-beta, which then facilitates the SMAD signaling. And SMAD4 actually also regulates GATA4, for example. And so when you lose this, so that explains why GATA4 itself is also downregulated. So the normal function of the 2A procollagen is to enhance a positive feedback control of the cardiogenic transcriptional network. And so I started off with this slide, which showed the extracellular matrix and all the different molecules in the extracellular matrix that you may have. And so I hope I've convinced you that the extracellular matrix does have a role in facilitating or titrating the availability and activity of growth factors. And in the case of the heart, 2A is a facilitator of the signaling pathways for cardiomyocyte differentiation and the gene regulatory network that ensures that you have proper cardiomyogenesis. And in terms of its applicability to humans, then we might actually see that in humans we have a very similar developmental mechanism, events that happen in human cardiac development. So, and the fact that we showed that 2A procollagen is expressed in the developing human heart, I would like to suggest that this is also relevant to the human situation. And so perhaps we might think then of variants in COL2A1 as being a risk factor for congenital heart defects, but also the application of 2A procollagen in promoting cardiomyocyte differentiation if you wanted to develop any in vitro assays to encourage cardiomyocyte differentiation. Perhaps you might like to think that 2A procollagen might help that. And so this work is done by a lot of people, and this is whoops, I should go back. This is the picture that we took. These are the most intimately involved people in my lab, and we were celebrating the paper acceptance, was very recently published. So we are very happy and thank you, but it was actually not possible without the collaboration of many people, and all of them really helped me in understanding cardiac development. Richard, Tim, and William, I think they were all very generous in sharing their expertise and their help. Thank you very much.
Thank you very much for Catherine Shah, Chair. Catherine Chair. Yeah, thank you. Any question?
Yeah.
Okay.
Wonderful story, Catherine. Wondering, actually, can you rescue the procollagen to a phenotype by locally overexpressing BMP2/4? just
can you repeat
by if you overexpress BMP2/4 in the cardiac crescent, whether you can rescue the procollagen 2A phenotype.
We haven't done that experiment.
But you would.
But the thing is, I think that would be also quite a difficult experiment to do.
Yeah, for sure. But I mean, whether it's also just simply accumulation there at the site, or whether it's also necessary for presenting the ligand to the receptor, I think that would also be very interesting to study. Wonder whether there is anything known. So you think it's primarily to concentrate then BMP2/4 these critical sites?
Well, I mean, I guess that's one idea is that perhaps it binds to the BMP. We know it binds. Then maybe together, and also 2A will bind, collagen will bind to beta-1 integrin. And so then the collagen would bind to the integrin, which then also that brings it close to the cell surface, which might then enable interaction with the BMP receptors, perhaps.
Kathy, wonderful talk. It's such a nice story to see where you started out, originally with this 2A and now looking into the cardiac developmental aspect. I wanted to ask whether or not you know that the collagen that gets made in that crescent stage then becomes part of the cardiac jelly that is formed when the linear heart tube takes place. Because for a long time, which I think we still don't quite understand why there's such cardiac jelly material, which is entirely ECM, and it clearly has an impact on development and looping as well as the valve morphogenesis, but whether the 2A is in that jelly.
In cartilage, type 2 procollagen, type 2 collagen binds to aggrecan, proteoglycans, which would make a jelly. I think they would attract water, so that would make a jelly. That would be one could be one explanation.
Okay. Yeah, no, because it's really interesting what the role for that jelly is, and it sounded like you are connecting with the presence of this procollagen 2A with a transcriptional regulating event, with the expression of, NKX. I guess maybe just as an extension to Thomas's question, is there separate signaling out of, the procollagen itself, besides from whether it is largely serving as a kind of a substrate carrier for other signaling molecule like BMP?
So actually, I didn't include this because it's not published, and we need to really validate it more. Interestingly, 2A procollagen also binds the nodal very early on, actually. We can actually show it in Western blots that it co-precipitates. And so we think, nodal and furin and crypto, they all form a complex, right? So this is probably, and then also Wnt signaling might come into it in the early stage. The other phenotype we see in the 2A null mice, which I didn't talk about, is because it's not relevant to here, is they actually have a head phenotype. There's a shorter, and they have holoprosencephaly, actually. So maybe a hedgehog is also affected. Wnt is affected. Yeah. So it's an important collagen.
And they're made by cardiomyocytes in that.
Yes,
early,
it's made by cardiomyocytes.
Yep.
It's not made in the second heart field. It's adjacent to the region where you would have the second heart field.
Okay.
Right. But it's made in the cardiac crescent.
Okay, great. Thank you.
Okay, thank you for your very interesting talk, Kathy. I also have a question for you about in your knockout mice, you have seen many genes downregulated related to the second half field development. Have you seen anything increase, such as the ALA2, whether it can compensate a little bit for the phenotype?
When we did this work, there wasn't single cell transcriptomics. There was in situ hybridization, so it was entirely on candidates. So we couldn't actually, it would be very interesting, I think, to do that now. Yes.
Okay, thank you.
There are Dr. Ne, Dr. Sugimura, Professor Wu, Professor Zhao, Professor Zhang, Professor She, and Dr. Tian. Please come to the middle of the stage for a group photo. Right, and then to the mobile phone. Thank you so much. Thank you so much. Please take a seat. Thank you so much, ladies and gentlemen. As we come to the end of the first day of the Culture Symposium and the YASS sub-conference, we would like to expand our heartfelt thanks to all the speakers, moderators, and participants for an inspiring day of exchange and discovery. For those who will be joining the banquet, please remember to gather in front of the entrance now, as we have arranged a shuttle bus which will stop by the Hotel Hyatt Regency Hong Kong Sha Tin very briefly to pick up other guests, and then we will head to the restaurant together. Thank you once again for being part of this meaningful gathering. So see you tomorrow and have a wonderful evening. Thank you so much, everyone.